Aloha and hafaday. I hope that we will get some more folks online. Thank you all for coming to our 2025 Summer Undergraduate Research Fellowship Final Symposium, co-hosted by the Pacific Islands Climate Adaptation Science Center and Hawaiʻi Sea Grant. We've had a great summer with our largest cohort yet, 12 students. We tried out a new workshop with our SURF alumni, and we have had our first student on Guam who's going to be presenting remotely. And we had an amazing group of mentors, many of whom volunteered. So I didn't have to go and recruit them, which was fabulous. We are excited to hear these presentations today about all of the great science that these students did over the summer. And there are a lot of them. So we're going to try and keep it to just 1 or 2 questions after each presentation, just to keep things moving. But let's go ahead and dive right in with some talks on plants and invertebrates. Our first speaker is going to be Allie.
Hello. Oh.
So Allie is a rising senior. You may come on up.
Okay.
Majoring in integrative biology with a concentration in environmental science and public policy at Harvard. Sorry, I went to Yale, so I have to say it that way. Or we have to like sign something at the beginning that tells, anyway.
Oh, I didn't know about that.
She focused on work this summer with Fred Rapun, Shimi Ri'i, and Alyssa Veneri at Hei'a Nur and on Nui Fisheries. So why don't you start us off, Allie? You can either use the arrows or the clicker.
Oh, okay. I'll do the arrow. Okay.
Is that gonna work?
Good.
There you go.
Okay. Okay. Hi, everyone. This summer, I was lucky enough to work with Alyssa, Shimi, and Fred as my mentors, and today I'll be sharing about my surf experience. So I would say my surf experience was a little bit more unconventional because I spent my time working on bits and pieces of several different projects. So in addition to working with my mentor, Alyssa, on her project involving Limu harvesting methods, I was also able to run my own parallel experiment as well as participate in La Lima workdays at Kako'oiwi. So first, I'll share about my experience working with Alyssa on her research, which focuses on the efficacy of different Limu gathering methods. Limu, or native macroalgae, are a culturally significant and ecologically important group of species to Hawaii. They are a traditional food source, a staple in the Hawaiian diet, and also are used in practices like ho'oponopono, or forgiveness ceremonies, in traditional medicine, and in hula. Limu are also primary producers, making them the base of local marine food webs. Limu also currently face several challenges, including competition with invasive alien algae, as well as other anthropogenic stressors like pollution, eutrophication, and overharvesting. There's also a loss of traditional knowledge surrounding Limu, making their preservation all the more challenging. So this is where our research comes in. Can we use and find traditional ecological knowledge surrounding Limu to both revive the cultural practice as well as support its growth? So this experiment focuses on 3 Limu species, Limu manawea, more commonly known as ogo, Limu palahalaha, and Limu kala. I forgot to put a picture on this slide, but Limu kala is the top and Limu palahalaha is the bottom. And Alyssa's looking at comparing 3 different treatments. So the first treatment is a control, so no harvesting or no disturbance of the Limu. The second treatment is the traditional harvesting method, which is very species specific and is catered to whatever the use of that Limu species is. And then the third treatment is the Department of Aquatic Resources, sort of one-size-fits-all method of collecting the entire Limu plant, except for the holdfast, so the part that attaches to the rock or the original substrate, whatever it was. And this is a picture of the setup. So there's about 1 gram of each Limu species attached to the bottom of these tanks, and they're being fed water from Honolulu Harbor because the setup is on, as Rachel mentioned, on Anuenue. Wait, did I say that right? Anuenue Fisheries Research Center on Sand Island. And after the Limu are introduced to the tanks, they're allowed to acclimate to the water for at least 1 week, and measurements are taken on a day 0, on a day 1, immediately post one of these 3 treatments, and then on day 14, allowing for a 2-week regrowth period after whichever treatment. And the way we measure growth in this experiment was through physical traits like length, dry weight, plant surface area, and effective quantum yield of photosynthesis or photosynthetic capability. And this experiment is still in its early phases, so some next steps include troubleshooting and continuing these experiment trials. And other exciting chapters include gathering Limu stories in an effort to collect more traditional knowledge, as well as the co-development of research with community members. And now I get to share about my own parallel experiment that I got to run, which will become a chapter in my undergraduate senior thesis. My thesis as a whole is looking at PFAS in Pearl Harbor, but this experiment specifically is looking at the uptake of PFAS in Limu. So to start off, what are PFAS? PFAS, or per and polyfluoroalkyl substances, are a large class of hundreds of chemicals. And you can imagine like a hydrocarbon chain, but all or some of the hydrogens are replaced with fluorines. So these man-made chemicals were made originally for consumer products because they have unique properties like oil resistance, being waterproof, being hard to break down. And obviously, these traits also make PFAS a harmful pollutant that's really hard to get rid of. And in fact, PFAS are now pretty ubiquitous in the environment and are recognized as a persistent organic pollutant. Sorry, there's so many acronyms. Persistent organic pollutant. PFAS are now linked to a multitude of adverse health effects like repressed immune response, developmental issues, and liver damage. And so why would we expect PFAS in Pearl Harbor? PFAS are often introduced to the environment through AFFF. Sorry, another acronym. AFFF is aqueous firefighting foam. And in 2022, there was a significant spill of AFFF in Red Hill, which, as we know, situations like this happen all the time. So I presumed that Pearl Harbor would be contaminated with PFAS. And you probably can't see the figure, but some preliminary water testing I did this past winter show that at these 3 sites, there are PFAS concentrations well above current health standards. So for reference, the EPA regulated amount of PFAS allowed in drinking water ranges between 10 and 100 nanograms per gram. And obviously we're not drinking Pearl Harbor water, but each of these sites had concentrations of PFAS of at least 500 nanograms per gram. And that last site all the way on the right, the USS Arizona Memorial site, had a concentration of around 2,500 nanograms per gram, which is just way beyond the 10 to 100 range. So to add on to that, my motivations for this project, Pearl Harbor is traditionally also known as Waimomi, which means waters of pearl. And if you know about Hawaiian place names, they're always descriptive and they always mean something. And as such, Pearl Harbor used to have things like pearl oysters and rich fish ponds and clean water and just provided sustenance to Hawaiians. So with this idea in mind, some questions I had or some goals I had for my project were restoring ecosystem health and incorporating traditional ecological knowledge along the way. And thankfully, I was able to work with my mentors on creating a project involving Limu that would help me achieve these goals. So my big questions were, can we utilize Limu in a way that will help bioremediate Pearl Harbor or help us take some of the PFAS out of Pearl Harbor's water? And so with my experiment, I hope to address the questions, how quickly does Limu take up PFAS? How much PFAS? And then most basically, is this Limu even safe to consume? So because this is a parallel experiment, the setup is very similar. I collected Limu kala and Limu palahalaha from Avavamalu, also known as Sandy's Beach. And my experiment also took place at Anuenue Fisheries Research Center on Sand Island. And that's a picture of my setup. I have each species of Limu in 2 separate tanks. But the critical piece of the setup is that the water from the tanks is coming from Honolulu Harbor, which I am going in with the assumption is also contaminated with PFAS. So in essence, I'm taking Limu from this clean water environment and introducing it to this polluted water and hoping to see some sort of uptake curve eventually. So I'm collecting Limu for PFAS analysis before introducing them to the tanks, immediately after introduction, and then periodically thereafter. And when I collect the Limu, I'm pressing them for to remove some excess water because what I really want to do is analyze the remaining tissues for PFAS. In addition to my PFAS extractions later, some next steps include doing more thorough water sampling across Pearl Harbor. So the top right is a map of my water sampling sites, and then data analysis to put it all together. And another aspect of my project that I'm still really trying to work on is communicating my results. I don't want stakeholders and community members to find out through like a random college undergraduate paper that their water is polluted. So I want to, of course, be respectful and maintain some sort of data sovereignty and deliver my results in a way that is useful and provides next steps. As for my hypotheses, I hypothesize that we'll see surge uptake. So I predict that the Limu will take up around the maximum amount of PFAS within the first 24 hours, which is what similar studies have shown. And observationally, Limu palahalaha seems to have a shorter life cycle. So I anticipate that the uptake will be quicker in that Limu species. In this project, I'm also looking at bioaccumulation factors, or BAFs. BAFs basically say by what factor does the Limu concentrate PFAS from its surrounding environment. So I anticipate that the BAFs will be relatively high, with Limu concentrating PFAS by factors in the hundreds, because other aquatic vegetation PFAS studies have shown similar numbers. And I also anticipate that Limu kala will have a higher BAF just because the tissues in it seem more dense, but maybe that's just a guess, I don't know. And if these hypotheses are true, this would mean that Limu has a high potential for bioremediation, but at the same time, this Limu that we use for this purpose would then likely be unsafe to consume often or in large amounts. So that leads to other questions I have. If we are to use Limu for bioremediation, what would we do with it afterwards? Because it would be unsafe to consume. And also, just for my project in general, can we use these BAFs to predict PFAS levels in other Pearl Harbor organisms? And last but not least, I'll be talking about my time in working in La Lima days. So in Hawaiian, La Lima means many hands, which is exactly what these days were. It was a time for volunteers from all over to come together in Heia, and we were working to fulfill Kako'o Oiwi's mission of restoring plentifulness and health to the land. In the moment, these days felt very separate from the experiments I was doing, but when I honestly was putting this presentation together, I realized that it kind of brought everything together for me. It made all of this summer cohesive. So besides being a break from working on projects or working on a computer, these days were, maybe this sounds corny and really sentimental, but it was a time for me to like remember why I got into science and research in the first place. Being someone who is part Native Hawaiian but didn't grow up super connected to my culture, incorporating traditional knowledge into science and serving native species and native ecosystems has been like my way of reconnecting and giving back. So to see so many people, so many hands come together to serve a similar purpose was really inspiring. And it reminded me of my motivation for everything I did this summer, as well as everything I hope to do moving forward. Yeah, okay. Thank you so much. That wraps up my summer recap. Thank you again to my mentors, Alyssa, Shimi, and Fred. And there are lots of people and places to thank, but to keep it short, thank you to everyone who shared their time and their space and their ike with me. Yes, I'll be taking questions. I know that was a lot, so thank you.
We have time for a question or maybe two, depending on how long the first question is. Anybody have one? Yes. Can you speak into the mic so that our online audience can hear?
Oh, Dr. Millikan.
Presentation. Quick question about PFAS and Limu. Are there studies or is there evidence that Limu are impacted as a species? Are they impacted by PFAS and in what way?
Like the organism itself?
Yeah.
There are some studies done on like the toxicological effects of PFAS on plants. I wouldn't say there's a lot done on aquatic vegetation, because when I was doing my research, a lot of the PFAS studies done on aquatic vegetation were actually for the purpose of bioremediation. So focusing solely on like, can the plant take up the PFAS and then remove it from the water? Not really like the effects that it would have on the plant itself, but there are like plant studies on more terrestrial plants that show that it does affect cell growth and all these things. But yeah.
Not good for anybody.
Yeah. No.
Thank you.
Yeah.
Okay, let's move on. Thank you.
Yay.
I see that.
Thank you.
Okay, Christine is next. Christine just finished her biology major at Manoa and has been working this summer with Blake Stone Osborne in Jeff Drazen's oceanography lab, analyzing zooplankton abundances in our island waters. Go ahead. Sorry, give me a shot. Yeah. And then you can either use the arrows or the pointer.
That fine?
Okay.
Hey guys,
Let us know if you can't hear her.
my name is Christine Uwaine, and this summer I've been interning at the Deep Sea Ecology Lab here at Manoa. With the guidance of my mentor, Blake, we've been investigating zooplankton communities along coastal offshore gradients. And let me start by orienting you on where we are. We are here in the Hawaiian Islands. We are right in the middle of a gyre, the North Pacific subtropical gyre, pretty much a downwelling zone, which just means there's like not much productivity going on in our offshore waters. So if you look into the gradient, the warmer colors just means that indicates that there's higher productivity. And on the other side of the gradient, the cooler colors, there's not much productivity going on. So if you look, if you look onto our oceans, there's not much productivity going on up until you get closer to our islands. You start seeing the warmer colors. And this phenomenon can be explained through the island mass effect, which basically says that near islands, there's higher nutrients due to several factors: upwelling, river outflow, submarine water, human runoff, and tall flushing, internal waves. And due to these increased nutrients, we have like higher phytoplankton blooms, and so like higher food available for so the island mass effect implies that islands support offshore food webs, but we don't know that for sure because it's kind of hard to study what these animals eat and their food sources. But just a little more background information. Zooplankton, they're the base of our marine food webs, right on top of or beneath the phytoplankton, depending on what the triangle is facing. And they get eaten by the zooplankton. Zooplankton gets eaten by the micronectons, which are small squids, shrimps, and lanternfish. And then they get eaten by the top pelagic predators, which are the tuna, marine tuna, swordfish, sharks, and dolphins, and all that. Just a little more background information. Zooplanktons are just those little tiny microscopic animals that drift along the ocean. And just thinking about it right now, as a kid, I would just, whenever I'm swimming, I'd take a big gulp of water. And I don't know how I feel about that right now after this internship. And yeah, there's many different types, just a couple to name. The top left, Brachyura, the first larval stage, and the second. Here are some copepods, the Cephalinidae. It's usually blue, so the way I would identify them is because sapphire is blue for identification purposes. We have some more copepods. There's a calanoid amphipod, pretty much looks like a cockroach. Quite abundant here as well. We got shrimps, gelatinous groups, and just a couple, but there's many types. Although zooplanktons definition, they're implied to be drifters, they actually have migrating behaviors. The first plot on the left top, that's an offshore behavior. During night, they would come up to the surface and their biomasses, they're on the surface. They eat at night due to, I'm sorry. And during the day, they would sink back to the bottom, the mesopelagic layer where there's no sunlight, barely any. That's due to, they're just hiding from the visual predators. Yeah. And then at nighttime falls again, they go back up to eat. because the food is in the surface. So similar behavior is happening near shore, the benthic emergence. The only difference from offshore and near shore is because we have substrates, we got places to hide, sand, sediment, corals, and at night they'd emerge up to eat, and during the day they'd hide. And another behavior, which ties along with the vertical, is horizontal dials, vertical and horizontal migration. So near shore, near islands, there's high food supply. So they would migrate vertically and horizontally, so like diagonally towards those islands to feed more food source. And so our research questions, our zooplankton community composition is different between near shore and offshore. Our zooplankton community composition is different in the same environment, daytime and nighttime, and any animal taxa to be associated or offshore. There's no high other airport. There's no light or what habitat type, substrate food availability, and more. We expect community composition to be different between day and night in the same environment due to benthic emergence, dial, vertical and horizontal migration. And lastly, we expect ostracods, mysis, cladocera, brachyura, and bivalves, echinodermata, stomatopods, and eggs to be reef associated. Some of these, I don't know if you've seen them at the beach, like the crabs, echinoderms, they're pretty much starfish larvae. And I don't know, but I would expect to see them near shore rather than offshore. So just some, so we had 3 different locations, Nanakuli, Waianae, and Makaha. That's to get an overall picture on what the leeward side looks like. So at each location, there's zooplankton toes, so just collection of the zooplankton. At near shore, which is a kilometer away from shore, mid shore 1 to 1.5 kilometer, and offshore greater than 20. So how it's done, the zooplankton ring, 1 meter zooplankton ring is connected to the boat. And we have this flow meter that's just to collect the distance. And as it gets pulled on by the boat, water gets filtered out and zooplankton gets collected into that collection tube on the bottom. And the net opening is a circle. So as it moves through a certain distance, it makes like a cylinder shape. So that's how we calculated our volume. And the height is just the distance the tow traveled. And that's just to standardize our calculations rather than just counting individual taxa. Let's see. And here's just behaviors we'd expect. Island associated, you see their numbers high during the day. Wait, no, I'm sorry. High near shore compared to offshore because they're residents of the island. And benthic emergence, they're emerging out of the sediments to eat at night. So you'd see their numbers increase with time. So as the day progresses from day, dusk to nighttime, their numbers start to rise because they're coming up to eat. Similar going on dial vertical migration, offshore, same thing. It's just the location, it's offshore, and their numbers are increasing as time progresses. So here's what we found. Here are what we found to be our island associated taxa. The first 3, ostracods, mysis, and brachyura, that's what we hypothesized to be island associated. And if you look onto the graph, the first one, the numbers are abundant near shore, and offshore not so much because they're island residents, and same pattern for the rest of them. For the benthic emergence, as time progresses, their number starts to increase, but they're the same ones as the island associated, and that's due to you'd find coral reefs where these sediments or these organisms rise out of to be near shore. So they're island associated, but not all island associated taxa are considered benthic emergent. If you look at this, the cladocera, it's not benthic emergent, but it appears to be abundant throughout the day. And maybe, I don't know, it has traits that, I mean, it's a little translucent, so I'm assuming that's how it's able to avoid predators, just to keep their numbers up. I'm not too sure. So plankton are pretty much understudied, so we can't tell you that. And diel vertical migrating taxa, these are shrimp. They're slightly larger. And offshore, their numbers are low during the day, and at night, they come out and eat, so their numbers are increasing. But if you look onto the left side, the nearshore, their numbers are also increasing at night nearshore. So they're doing that vertical and horizontal migrating. They're also moving closer to the island because they want to eat better, more food. I don't know. So what we did not expect, what we expected to see is eggs, echinoderms like starfish, sea cucumbers, and sea urchins, stomatopods, they're like mantis shrimps, to be near shore. I feel like if you go snorkeling, you see a bunch of these. But their numbers were also abundant offshore. So maybe currents push them along, or we just need more toes, more data, better conclusion. Okay. Relative abundance. There's not much on our y-axis, that's the relative abundance, and the x, that's the toes by each station. And if you compare nearshore and offshore, there's not much difference except for the forms. And so there's not much of a community composition difference nearshore versus offshore. Whereas if you compare each station, this is for the Makaha near Nanakuli, they're different, and that's due to the benthic emergent taxas and the diel migrating taxas, also nearshore and offshore, different guys showing up to eat at night. Here's another plot of PCA. It just shows you if communities are different, then they would be separated. And by color, nearshore and offshore, those communities are pulled to different directions. And by shape, it would be time. They're also pulled in different directions. So what this plot is telling you is the communities differ temporally and spatially. The next step. So what we just did, the DNA barcode, no, we didn't do the DNA barcoding, we did the cell scan. And that gave us an abundance, so a number, but it didn't tell us who these guys are, because we're working on family and order, like a very broad category. So in order to figure out who these guys are, we would have to do some meta barcoding to get us into species level. And in the fall, Blake would be analyzing micronectons. These are the predators of the zooplankton, and to see if what's inside their gut content is the reef associated, and if it is what's considered the reef associated taxons, then there's that trophic link between offshore food webs and islands. And this is very important to us because we rely heavily on the pelagic predators, tunas, swordfish, spinner dolphins, ecotourism. Our diet's mainly composed of it. And so if there is that trophic link, that means islands are supporting these offshore food webs. We are supporting, well, they are supporting these larger, and we must protect our reefs and our coastal and habitats. I don't know about you guys, but if these guys start to disappear, and I don't know, no poke, I probably would disappear alongside them. And yeah, I wanted to give a big thanks to everybody here, Rachel Sea Grant, PyCast for funding this opportunity, and my mentor, Blake, for your guidance and support. Very grateful. Thank you.
Great. Do we have any questions? Oh, wait, we need a mic.
Great talk. That was so awesome. I love plankton. I saw that you had some forams in there, my favorite. I was wondering, so you mentioned that the eggs and echinoderm larvae and the mantis shrimp were kind of surprising because you didn't expect to see them both near shore and offshore super abundant. Do you have any thoughts about why that happened? Is it because like they're dispersing, or they have some protection on them that lets them not get eaten, or like what's up with that?
What I think is we didn't take account currents. Maybe they got pushed along, so offshore. So that's what I would think.
Oh, cool. That's awesome. I'm super curious about the eggs, so you're giving me a lot to Google later on. Mahalo.
Great. Thank you. I just recently did a manta ray night snorkel, and the plankton were abundant when we put those lights on. So I guess it was all the water was cloudy until the mantas came up and just ate them all.
Oh.
And then you'd get these moments of clear water and you're like, oh, it was cloudy because there were plankton. Okay. Anyway, it was very fun. Thank you. Okay, let us move on then. Next.
Taryn. Is she going first? She's going now.
Who is that? Okay, just to remind folks online, please keep cameras and mics off during the talks. Thank you. During question time, you can turn them on if you want to ask a question. Okay, Taryn is a rising senior who's majoring in marine science at Hilo. So she flew over this morning. Thank you for coming. She's been working with Steve Dew this summer, looking at the role of sea urchins in reefs. So take it away, Taryn. Oh, I know, because I didn't click. There we go. Okay. So you can use that with yours. Okay.
All right. Aloha, everybody. My name is Taryn. My project title was Assessing the Bioerosion Rates and Excrement Matter of Trypnosophis gratilla and Echinometra mathii. So to kind of give you guys a little brief overview before I dive into my project, I wanted to talk about two very important ocean processes. So I want to talk about accretion and erosion. So accretion is the buildup of material. So that could be coral, sediment, algae, things that build up our reef. And erosion is on the flip side of that. So things that break away, such as storms, currents, and even natural things like sea urchins and parrotfish. And in a healthy reef habitat, we're going to want to have more accretion. So we're growing instead of erosion more. And if in some cases we have more erosion than accretion, we can see a loss of biodiversity and the reef weakens. And here you can see at this left photo, this is kind of like a healthy rate of accreting. You know, our coral reef is healthy versus on the right side, the reef may have heavily eroded and not been able to withstand other problems such as pollution and ocean warming and ocean acidification and things like that. So a little background on urchins, which is what my project mainly looked at. These guys are primary bioeroders, and they typically mainly eat algae using their little mouth. It's called an Aristotle's lantern, and it's kind of shaped in a beak, and they just kind of use it to grasp material off. And due to the fact that they typically only eat algae, a place called the DARD, the Division of Aquatic Resources in Hawaii, actually used this knowledge and started to grow the Trypnosophis gratilla in an urchin hatchery in order to deploy them in places where the algae populations were too high and where the algae was outcompeting the coral. So using this as an advantage to maintain that balance of algae to coral. On the flip side of that, there was a study done actually on the Big Island looking at fauna populations, which are the super scary big black urchins, and looking at their population density because it was extremely high, higher than any other place that they had seen globally, and looking at their effect on the coral reefs there at the Honolulu Bay. And they actually saw that the fauna was decreasing the carbonate budgets, and therefore they were grazing over the coral and eroding that coral. So in cases like this where we might have few natural predators of these urchins and that causes that spike in urchin population, and paired with a low algal density, we can see that the reef structure erode. So a little importance about my project. So these specific urchins, the Trypnosophis gratilla and Echinometra mathii, also known as like collector urchins and rock-boring urchins. So the effects of these species have not been looked at in Hawaii. So just seeing, you know, what this does at our local reefs, also super beneficial in conservation and resource management decisions. Just helping in predict, you know, what reefs are at risk, and maybe do something like DAR and implementing more urchins in cases where the algae population is super high, or even saw in the Haudenosaunee to the our reefs. So my two research goals were to quantify the release of the Triphysis scutellum and also find 10% of the wild urchin excrement is constantly carbon. So a little background on my location, kind of where I did most of my lab work was at PACRAC, the Pacific Agriculture and Coastal Resources Center, located on Big Island. Super thankful to work in the coal mystery section. We use multiple sections of the resources center, but I was thankful to be in this section because I worked with this mentor, and he had the forefront of very much like community-based helping an event. So I was thankful to be able to work with up or down high school students and teach them processes, working with another internship program called Porites, and so working with them. And then my personal favorite, we had a welcome event with the Kyoko community and just showed them what we do on experiments in this field. So super thankful for that. But diving into my project, so I collected my urchins and substrate at two different locations. Here on the left is the Chalks Beach, and then and I was collecting two different types of substrate. So I was looking at lava rock and calcium carbonate rock, and then also looking at the two different species, so the Triphysis scutellum and Macchiayi. And then a lot of my time was designing and creating these pages that you see here on the left. I like to call them my urchin jail cells because they're made out of chicken wire and PVC pipe. And essentially, I wanted to keep...
Rachel, can you mute Helen?
Okay. But essentially, my urchin gel cell is made out of chicken wire and PVC pipe, just designed to keep that urchin with it, so it didn't have any plus, you know, what effect it's having on the certain substrate. So yeah, that was kind of my setup. And then for data collection, I did weekly buoyant weighing on those rocks just to see, you know, are they losing weight over time? How much are those urchins eating? And then also taking photos of my rocks and also my urchins at the beginning, in the middle, and the end. And then for the urchin excrement part of my experiment, so this was a very short time where I was collecting data, but essentially I just went out, collected my urchins, and then put them in the jars that you see on the far right. And they were placed in that jar with a bubbler, of course, for 24 hours just to collect their fecal matter just in that time frame. And then after the 24 hours, I took them out of the jars and filtered and dried the fecal matter. And then I actually added, after it was completely dry, I added a mixture of 10% HCl in order to dissolve whatever calcium carbonate that was in their fecal matter. And so then that way I could determine, you know, what percent that they're actually grazing from the corals. So into my results, my first graph, this is the rubble weight change over a 4-week period. I just kind of wanted to give a timeline and for you guys to see how they slowly decreased over time. So here on the x-axis, I have week 1, week 2, week 3, and week 4. And then on my y-axis is the weight loss. And I also colored by lava rock and calcium carbonate. And as you can see in my graph, the majority of the weight loss came from the calcium carbonate. So they lost more weight over time, and then lava rock didn't lose as much. But they were still gradually decreasing over time, showing that those urchins were slowly eating at that substrate. And then this is kind of the average weight loss over the 4-week period of time. So here on the bottom, I have the trypnoces on the left and the echinometra on the right, and then the weight change here on my y-axis. And I did it final minus initial, which is why it's in the negative. This is kind of what they lost in that 4 weeks of time. And I did find that, and as you can see in the graph, there was a statistically significant difference between the substrate. They preferred it over the calcium carbonate over the lava rock. Unfortunately, there really wasn't too much of a difference between the two species, but I did find that they did like that calcium carbonate more than the lava. And then into my urchin excrement data. So this is just pure fecal matter dry weight. And so here on my x-axis, I have the echinometra on the left and trypnoces on the right. And then the average fecal matter here on the y-axis. And this kind of graph just shows you that the echinometra did not produce that much fecal matter. So they're a lot smaller in size, which makes sense. And the trypnoces had a lot more. And so just kind of keep this graph in mind when we move on to the next one, which is our percent carbonate in fecal matter. And again, just the echinometra on the left, trypnoces on the right, and percentages. So they had fairly about the same amount percent carbonate in their fecal matter, which is interesting to find because I definitely thought with the trypnoces being larger and producing more, I thought that they would have more percent carbonate, but they actually were eating about the same amount, which was interesting. But some future directions and reflections, some future pathways for this experiment. I would love to run this experiment on different species, just because different species inhabit different niches in the environment and just kind of looking at an overall effect on this reef ecosystem. And then also doing more urchin excrement analysis because my sample size was pretty small and I got some cool data from it, I feel. So I would love to run that a few more times. And then some reflections. I've always loved urchins, like going to the beach. I would always flip over rocks and play with them. But after starting this experiment, I was like, okay, wait, I really don't know much about urchins. And so I was super thankful to kind of dip my toes in the water and really look at their effects on the reefs in Hawaii. And also my favorite reflection from this was just getting involved in community-based work and really putting that at the forefront of my mind moving forward in my scientific career and maintaining those ties. And I remember interviewing for this position and telling Rachel, guys, I really want to give back to this community that I feel has shown me so much aloha. And so I was super thankful for this summer because I feel like I was able to do at least a little bit in that aspect. Yeah, thank you.
Do we have any questions for Taryn? Urchins out there? Yeah.
Just one. Oh, thank you. I was just wondering what your favorite part of this entire process was, because it sounds like you planned out a lot of this, and I'd like to know what you had the most interest in.
The most interesting? I would say my favorite part of the experiment, though, was our internship was definitely engaging with the community. I think talking with aunties and uncles from the Keaukaha community, and they were like, I've driven past this every day of my life and I didn't know what you guys were doing this whole time. And so being able to share that with them was just amazing. Yeah. So that's my favorite part, I think.
Great. Are there any other questions? Okay.
They're so fun.
I know you're an invertebrate kind of.
I love invertebrates. I was wondering if you have thoughts about why the urchins like the calcium carbonate substrate over the lava. Is there something growing on it that's different?
Sure. I think it's because it might be easier for them to chew on because lava rock's super tough. But yeah, that would be my only hypothesis, was that it's just easier for them to munch on with their little mouths.
Yep. Mom.
Okay, well, thank you.
So thank you.
And we shall move on. Oh, that was a cool picture. Yeah, some of those really shrank, didn't they? Anyway, okay, Allie, hi. Let me click that on somewhere. So Allie is a rising junior in marine biology at UH Manoa. And they worked with Melissa Melendez this summer, focusing on corals. Allie helped with experiments on mitigating ocean acidification. Show us that element of this. Thank you.
So hi, everyone. Thank you so much for being here today. So today I'm going to talk a little bit about my project, which was understanding the effects of increased total alkalinity on the coral species Porites compressa. So first, a little introduction. Hi, I'm Allie. I'm a junior at UH, currently pursuing my Bachelor of Science in Marine Biology. I was one of the Pacific Island Climate Adaptation Science Center scholars, and my mentor was Dr. Melissa Melendez, who is here with us today. And I was able to complete my internship at the Hawaii Institute of Marine Biology at the island of Moko'olo'e as part of the Point Lab. So first, a quick background on some concepts that are kind of integral to my experiment. Ocean acidification is the reduction of ocean pH from an increased concentration of dissolved carbon dioxide. This is coming from increased outputs of carbon dioxide into the atmosphere from anthropogenic causes. As you can see in this graph, it's only been increasing. This just shows the past about 25 years, but it's really been increasing ever since the Industrial Revolution. And this is a problem because the ocean absorbs about 30% of this atmospheric CO2, with May 2025 being an all-time record high of atmospheric CO2 that NOAA found. So the actual chemical process of acidification happens when this increased amount of carbon dioxide comes down into the seawater, pairs with the water molecules, and forms carbonic acid. This carbonic acid then dissociates into a hydrogen ion and a bicarbonate ion. And this is kind of where the problems arise, is because this free hydrogen ion wants to pair with carbonate ions, which naturally occur in the ocean. And this is an issue because carbonate ions are one of the main building blocks for calcium carbonate, which a lot of organisms such as coral use to, build their skeletal systems. So because the hydrogen ions are taking this carbonate ion away, they have less resources in order to build themselves with. And this causes their skeletal densities to decrease, and they become a lot more brittle and easier to break. Now, alkalinity is the buffering capacity of water, or the ability for water to kind of maintain a more stable pH. And this could be useful if we're able to kind of bolster the ocean's alkalinity, then we can make them less susceptible to these changes in pH. This study in Australia was actually done and found that on a species of plate coral, the top row you can see there is the coral that was in the control group, so the less amount of CO2, and you can see that the skeletal structure is a lot stronger, a lot more uniform, versus the bottom, where a lot more ocean acidification has taken place, and the skeletal structure is a lot less uniform, you could just look at that, you could just break that, so not good for the corals. For my specific experiment, we were looking to increase the kind of target alkalinity to about an extra 200 micromoles per kilogram above ambient and natural conditions, because coral reefs, especially in Kaneohe Bay, have about a change of plus or minus 100 micromoles per kilogram. So in order to get around this kind of window of variability, we decided to do it an extra change of 200. So for some of the chemicals used, the minerals used to change the alkalinity, in the broader experiment, they were using sodium hydroxide. However, I did not use this in my personal experiment, but it is a common mineral byproduct of a lot of electrochemical processes. The two minerals that I used for my experiment, however, were calcium oxide, which is an industrially produced mineral, and olivine, which is more of a naturally occurring silicate mineral. So my hypothesis overall was that by increasing the total alkalinity of seawater, Porites compressa growth and net calcification will increase. Going on to my methodology, I chose the coral species Porites compressa because it is the dominant coral species in Kaneohe Bay where the lab is located. So it's a good indicator of kind of a lot of ecosystem health. And I chose 3 larger colonies from the bay, and then I took 9 fragments per colony, and I affixed them to these little plugs. They're about the length of a finger. So I had a total of 27 fragments, and then I put 3 fragments from each colony into each treatment tank called a mesocosm. And my main things I'm measuring again were net calcification rates, which is kind of a coral's short-term response to these treatments, or how fast they're able to kind of cycle through and take in calcium carbonate and build their skeletons. And I was also measuring net growth by buoyant weighing, as you can see on the bottom there. That was my little setup, where I weighed at the beginning of the experiment and at the end of the experiment. And this is more of the long-term response that each coral underwent from the treatments. So as far as how the treatments actually worked, as you can see at the left here, there's the Kaneohe Bay water coming in, and then it's getting mixed with each respective mineral. And then the minerals are then dispersed into each mesocosm or treatment tank to be introduced to the corals, and then the water then flows out back into the bay. So this mesocosms are an outdoor system of tanks in order to mimic the environment under more controlled conditions. And as you can see, we had a schematic of about 12, and there's 3 per treatment. But again, I did not utilize the sodium hydroxide, so I used the 9 of the 12 mesocosms. Each mesocosm also, obviously I was operating under the larger experiment going on. So there were 20 samples of Montipora capitata and 20 samples of Acropora acuta, two other common coral species in the bay. This was helpful because it helps to simulate more of an ecosystem environment and see how the corals can maybe interact with each other as well, instead of just being isolated. And the little square here is the positions of each coral, and they were interspersed with each other. So in order to measure my net calcification rates, I utilized a closed jar incubation using chambers. I used about a liter full glass jar, and I filled it up with water, and then I put in these little stands for the corals to sit on with zip ties and PVC pipe, very high tech, I know. And each coral would sit on top of the egg crate. I would then fill each jar with the water from each respective mesocosm and close it off with some parafilm on top, let it float around in there, as you can see on the bottom right, and I would let it cycle for 2 hours. I did this twice over the course of the experiment. Unfortunately, I could not do it on all of my coral fragments, so I just did it on one fragment from each treatment, all from the same colony. So I would take an initial reading of the water inside of the mesocosm, and then after it had cycled around for 2 hours, I would take the jars out and take another total alkalinity reading from inside the jar, as well as a buoyant weight of the coral and quantity. So why did I take all of these measurements? Well, this treatment from one of our PX lab product vehicles. On the CFC water, final and total initial total alkalinities, volume of seawater, time distributed, and alkaline right weight each fragment. So because I obviously took buoyant weights instead of dry weights, I just did version theta using this equation, and then I was ready to calculate my net calcifications. So going on to my results, first with net growth, or the kind of long-term response of each coral fragment. This is divided into both colony and which treatment the fragments were a part of. So each of these bars represents 3 different fragments. So you can see it definitely varies between colonies quite a bit, but if you compare each colony graph for each of the same color bar, you can see some differences in growth, with calcium oxide proving to be one of the treatments that resulted in a little bit more growth, and the control being the second most. Now for calcification, I did this twice over the course of the experiment, like I said, so you can see the blue bar on the left here is the first time I did it, and the pink bar on the right is the second time I did it. Something I thought that was interesting is that if you look over at olivine, the first time around I did it, it proved to result in the highest net calcification rates, but the second time I did it, it was actually the lowest. And I was wondering, like, oh, why would that have happened? But then I was thinking and realized that towards the end of our experiment, we had a little bit of our olivine concentrations being off of our seawater treatments. So I remember looking at the total alkalinity readings and them being lower than expected. So that could be a reason why the second time around they didn't show higher net calcification rates, is that our olivine treatment was like a little off. And this is just kind of that graph consolidated into the overall treatments. So as you can see, olivine proved to be the best. So overall findings, preliminarily, calcium oxide fragments had the greatest growth or the best long-term responses. However, they were not statistically different enough from the control given the time I had to make any conclusions necessarily, but it does show promising preliminary results. And also preliminarily, olivine fragments showed the greatest net calcification rates. Again, not statistically different enough from the control. But something I thought was interesting was that the calcium oxide and olivine net calcification rates were statistically different from each other, maybe pointing to calcium oxide making it harder perhaps for these corals to calcify. But overall, it did point to ocean alkalinity enhancement treatments possibly improving overall coral activity. It's just different minerals seem to have different effects. So in terms of future work, more research should definitely be done into this topic to investigate both the growth and net calcification rates of corals, because just given the limited time I had, I only had 24 days to complete this experiment, and just the amount of replicates I had, this definitely could be repeated in a more effective way. but it still showed very promising results. And then more things that should also be studied are definitely other ecological responses and ecosystem responses with ocean alkalinity enhancement. Not just other coral species, but other calcifying marine organisms and even non-calcifying marine organisms, because if we want to use this as a mitigation technique, we have to make sure that before we put it out into the ocean, we understand everything that could possibly happen from it. So just a couple other fun stuff I got to do this summer. I was kind of everywhere. So my daily duties at the island included taking a lot of the total alkalinity readings per day to just make sure our treatments were looking like they should be, taking assessments of the kind of salinity and dissolved oxygen levels of each tank, buoyant weighing a lot of our corals, and even taking microbe samples of each coral, which was something we weren't expecting to have to do. But unfortunately, into our experiment, we found that our Montipora corals had a strange, we thought it was a mystery disease, and we weren't sure what was going on. They were paling a lot. There was a lot of soft tissue kind of breaking off. So we took a lot of microbe samples from each one to see what was going on. Unfortunately, we did have to shut down the experiment a bit early because we weren't sure what was going on, and we didn't want to potentially contaminate the bay with what it was. Luckily, we found out later it was just a flatworm infection. So nothing to be too concerned about, but we did end up having to shut down the experiment about a month early. And other things I got to do were some more field work, deploying map CO2 buoys, looking at how this is an example of one of the automated sensors we're looking into to take total alkalinity levels of the bay and look at overall bay health. I got to take bay samples, and then I got to come back into the lab and analyze them myself. So the bottom left is the dissolved inorganic carbon machine. It gave me a lot of trouble, but we were able to make it through some samples. I was also able to prepare a lot of samples for nutrient analysis, as well as looking into different just lab kind of methodology. So I just want to say a quick thank you so much to my mentor, Dr. Melissa Melendez, the PI for our lab, Connell McNichol, Dylan Bowman, the UH lab tech, Eleanor Tenbrink, Raina McClintock, Naomi Rohrbach, and Amy Markle, all the grad students in our lab, and Zach Quinlan, one of the postdocs in our lab. Without all of your help, I truly could not have gotten anywhere I did. So I just really want to say thank you, and probably supporting me and answering all my stupid questions. So thank you so much. And of course, big thank you to Rachel Lynn, the program coordinator, and all of my fellow surfers and the program as a whole. And PyCast, without any of this funding or opportunities, I would not be where I am today. So thank you so much.
Very cool. Do we have any questions? I apologize for the speaker. We've been having periodic problems and it's electronic, so it's really hard to pin down. But anyway, go ahead.
So did that month where you had, did that affect your data a lot, or like more data, and maybe that would have affected your final results?
Yeah, so I definitely, at least for the net calcification, I wanted to do that chamber process at least one more time, just to have 3 instead of 2. And with that, we probably would have been able to see more conclusive data with just basically doubling the experiment's time. But unfortunately, you got to do what you're given. So it was still a very valuable experience and lots of cool stuff. So.
Sorry, I have one more question. What would you have expected the results to maybe look like if you had been able to take that final amount of data?
I would definitely expect to see more of a difference in the growth rates. So like I said, they weren't statistically different enough to draw a conclusion, but we would see more of a difference, like more of an increase in the calcium oxide growth or more of an increase in the olivine calcification.
Got you. Thank you, Allie.
Thank you.
Great, thanks. Okay, so from there, we are going to go over to Guam. I hope Bianca is online. If you are, please, yes, there you go. Let me unspotlight this so that we can see you. And I'm going to spotlight you. Okay, so let me introduce you. Bianca is a rising sophomore doing a chemistry biology major at the University of Guam. She worked with Lori Raimundo in the UOG Marine Laboratory this summer. Bianca is also exploring corals and focused on cultivating at-risk, I'm going to mess it up, Acropora corals She's going to be sharing from Guam. So if you are ready, you can go ahead and steal the sharing. I'll stop here.
Got it. Thank you.
That end.
Can everyone see?
Yes, that looks great.
Okay.
Thank you.
Okay, so I will start now. Half a day, everyone. My name is Bianca Borja, and I am an undergraduate student at the University of Guam. And for today, I will be presenting my SURF project titled Initiating Ex-situ Culture of At-Risk Staghorn Acropora Using Micro-Fragmentation. This work was conducted under the mentorship of Dr. Lori Raimundo from the UOG Marine Laboratory. Before I dive into my research, I'd like to give a little background on Guam's coral reefs. Between 2013 to 2017, our island went through a series of mass coral mortality events. High sea surface temperatures caused bleaching, extreme low tides caused subaerial exposure mortality, and also disease outbreaks. As you can see from the pictures on the other side, bleaching and diseases like white syndrome severely damage our coral communities. Next slide. And these events then led to the idea of coral restoration. The island's marine management agencies recognized the need to develop ecological restoration techniques. The PD Baumholz Marine Preserve Coral Ocean Nursery was then established in 2013, and they were developed to provide refugia for species at risk of extirpation, provide a culture stock of corals from surviving populations for outplanting, and also to preserve genetic material. Coral nurseries have now become a well-established part of restoration efforts in Guam, but a new idea was then introduced to start cultivating Acropora corals in an ex-situ setting, and this leads to our objective. This project focuses on initiating cultivation of at-risk staghorn Acropora coral species in an ex-situ setting using aquarium-based systems. And 7 species of Acropora were taken from the PD Baumholz nursery for this project. And these 7 species include Acropora muricata, acuminata, aspera, teres, vani, austera, and virgata. I will then answer these 3 questions for this project. How did the 7 species perform relative to each other? How did the 7 species perform in closed versus open systems? And how did the 2 positions, upright versus recumbent, influence early growth? And for the last 2 questions, they will come up naturally as I present my methodology. Moving on, my methodology. For this research, I introduced the 7 species to 2 culture environments, this blue room, which is a closed system with recirculating seawater, and the lanai, which is an open system using a flow-through system with water from Pago Bay. 4 fragments from each 7 species of Acropora were fragged, and 2 fragments were each assigned one of 2 orientations, either upright, which we will refer to in the data as vertical, or recumbent, horizontal. Each fragment was glued to a ceramic 2 by 2 inch tile with superglue gel, and a total of 84 fragments were observed, 42 being vertical and 42 horizontal. As you can see for the pictures, the blue room is a closed system, and the lanai is an open system. And for the orientations, recumbent is basically the coral laying flat, and for the upright, it's just standing up. Okay. For our tank environments, so I had to measure the tank environments, and for the temperature, I used an onset hobo data logger, and for the light, I used a PAR sensor. I also conducted daily health checks, which included checking for pigmentation and signs of disease, and also start of basal growth and tissue resheathing over the cut edge. Growth measurements were done every 2 weeks, and for my fragments itself, I didn't have the same start fragging dates for all the species. So the measurements was taken 4 or 6 weeks. And for the growth measurements, I measured the main axis growth, which is the length of the coral, and also the basal growth. And these measurements were analyzed using ImageJ. And again, the photographs were taken every 2 weeks. And I also counted for the appearance of apical corallites, which signify new branches. And aquarium maintenance was also done. Tanks and the fragment substrate were cleaned every week, and dead fragments were removed.
And from the pictures, you can see me taking pictures of my corals, my baby corals. Moving on to the results, I wanted to first highlight the environmental differences between the blue room and the lanai. For temperature, the blue room stayed quite stable, ranging from about 26 to 26.6 degrees Celsius. As for the lanai, it was much more warmer, ranging from 29.5 to 30.5 degrees Celsius. When we look at the light available for photosynthesis, which is considered as PAR or photosynthetically active radiation, for the lanai, it ranged from 0 to 200, and for the blue room, it ranged from 0 to 175. Moving on to our next slide, which is survivorship. This is just the number of coral fragments that survived during the study. From the table, we can see that the blue room had no coral mortality, as seen right here. Every fragment survived, and in contrast, the lanai had 38 out of 42 fragments survive. Looking more closely at the species, the fragments that died in the lanai was the Muricata right here, and the Vani right here. Moving on to the next slide. So this slide covers mean apical corallite, which refers to, again, the formation of new branches on the corals. From the table, we can see that overall there wasn't a lot of new branches that grew. However, 5 of the 7 species did show some apical corallite growth. Growth tended to be slightly higher in upright fragments and in those kept in the blue room. As you can see from the pictures, you can see the little branch growths over there. And these are called apical corallite. Next slide. These graphs show the differences in total coral growth based on environment and fragment position. And just to summarize, the graph on the left represents the main axis growth. Here we can see that in the blue room, the blue room fragments grew more in both vertical and horizontal positions. And the graph on the right shows the basal area growth. And for the fragments in the horizontal position, the lanai actually had greater basal growth, while for the vertical fragments, the blue room showed higher basal area growth. Moving on to the other one. So this graphs, it's just the total main axis growth by species and fragment position. In the blue room, this graph. Almost all species grew more in the vertical position compared to horizontal. Austera showed the highest vertical growth, followed by Aspera. And for horizontal growth, Aspera grew the most across the species, though it was still less than its vertical growth. As for this graph, in the lanai, the trend was similar. Vertical fragments generally grew more than horizontal ones, and Aspera had the highest overall growth, particularly in the vertical position. And you may notice the large error bars. It just indicates that there is high variability, and this is due to the fact that I did not have a large sample size for the species that I was working with for this project. Moving on. This is just an example of the growth that can be observed from the 4 to 6 weeks that the corals were in the project. As you can see, there is an observable difference in length for the vertical and also for the horizontal. Next slide. Okay, so this slide looks into the basal area growth by species and position. This one, the blue room graph, vertical fragments consistently had higher basal growth across all species. Again, Austera showed the greatest basal area growth in the vertical position, while Aspera had the highest basal growth in the horizontal position. In the lanai, this graph, Austera had the highest total basal growth in both vertical and horizontal positions. However, unlike the blue room, there wasn't a consistent pattern of vertical versus horizontal growth across species in the lanai. And again, to give a little example of the basal growth, vertical right here, you can see that from the beginning, there wasn't a pronounced basal attachment, but for my last picture measurement, you can see that there is tissue that is starting to grow on the substrate. And as for the horizontal, you can see right over here that there is sign of basal attachment. And that is basically what I had to measure for to graph those graphs. Okay, color difference. So this graph shows the average color differences of corals in the blue room and the lanai. Using the color code guide, which ranges from D1 for lightest to D6 for darkest, I calculated the average color for each species. And from the graph, we can see that corals in the lanai generally had darker pigmentation compared to those in the blue room. And this difference is also visible in the photos right here. So in the blue room, it can be seen that it's a little bit more paler, and in the lanai, it's a little bit darker. And that is it for my results slides. I will now move on to conclusion. So for my conclusion, I answered 3 of the questions that I proposed in the earlier parts of my slides. So for species performance, overall, Aspera from the lanai and Austera from the blue room showed the highest mean total main axis and basal area growth for mostly vertical positions. And for my lowest performing species, Ader showed the smallest growth across all environments and positions. Second question, blue room versus lanai. Corals growing in the blue room show higher growth across all species and positions. There was a consistent observable difference in pigmentation between the 2 environments. This requires more study because it reflects differences in endosymbiont density. And it can also be concluded that light regime in the blue room requires adjustment. And the blue room provides more stable conditions than the lanai environment. Third question, upright versus recumbent. Upright fragments grew faster than recumbent fragments across almost all species. Recumbent fragments stabilized on the substrate more quickly, and upright fragments had slightly increased apical counts, and these include Austera, Mercata, and Aviar. There did not appear to be an advantage of growing the fragment in a recumbent position unless you would want a slower growth rate if you are planning to establish a collection in an aquarium setting. And my acknowledgements. So I would like to thank PICAS Pacific Islands Climate Adaptation Science Center for giving me the opportunity to take part in valuable research. I would also like to thank National Fish and Wildlife Foundation and also the University of Guam Marine Laboratory for giving me the space to conduct this research. Special thanks to Dr. Lori Raimundo, the best mentor ever. Mike McHugh, best aquarium man ever. Dr. Rachel Lentz and John Borja. Thank you, John, for taking the best pictures ever. To Meg, Ashley, Vivian, Farron, and Emma, thank you very much for helping me frag and also stick the fragments on the tile and just the logistics of it all. Thank you very much. It wouldn't have happened without you. And also special thanks to Keanu and Janelle for providing education on the drone core availability here in Guam, and also for this wonderful picture that I have been using throughout the slides. They actually took that picture of the PD bomb holes. And that is it for my presentation. Thank you very much.
Thank you, Bianca. That was great.
Does anyone have any questions?
We have time for some questions either here in the room or online. I keep forgetting to say if there's anybody online who has a question, they can pipe up as well. Have any in the room? We do have one. Hang on, getting a mic.
Thank you. My name is Jenny. I'm really interested. I studied symbiosis, but I'm really interested in the pigmentation that you saw and that potential for increased symbiont density. Do you want to speak more to what you think you might find there, that perhaps those growing on the lanai, would you consider them to be healthier than those growing in the blue room, or what would you say about that?
So I wasn't really able to explore a lot about the pigmentation for the lanai and the blue room. It's just an observable thing that I was able to see, that it was a little bit more paler in the blue room than in the lanai. So I'm not able to conclude much from the pigmentation itself, but it's a aspect of you're taking care of corals. It's an aspect that can be explored in the future. And there's another question in the chat. I will just read it. What did you enjoy most of your work? I enjoyed snorkeling, and I enjoyed going to work every day to check on the corals. I basically named them. And that is it.
Okay, then I guess we are good. Thank you, Bianca. Okay, we are exactly on time. Absolutely remarkable. So you now get the reward of a biology break. You may get up and stretch and hit the head, whatever you need to do. Do we need 5 minutes or 10 minutes? Okay, we're going to do a 10-minute break, and we will start back promptly at 1:55 with Mariela. So thank you all. And don't just leave, come back. Okay, so we're going to be starting the next chunk of talks, and these are kind of high-techy sort of talks. Mariela is a rising sophomore doing marine science at Hilo. She worked with John Burns and the Mega Lab this summer, learning how to do some 3D mapping and analysis of Opihi distribution, among other things. So go ahead, Mariela, and tell us about what you're doing.
Alrighty, thank you so much for the lovely introduction, Rachel. As you just heard, my name is Mariela, and today I will be telling you about what I did over the summer, which was intertidal mapping, living life on the edge. So first, I'd like to introduce that welcomed me into their arms, the Multi-Scale Environmental Graphics Analysis Lab, better known as the Mega Lab. As you can see, scientists love their acronyms. Mega Lab is no exception. And if you haven't heard of them before, the Mega Lab is composed of incredible individuals who are not only dedicated scientists, but creatives, such as surfers, artists, and filmmakers, who use their skills in tandem with their work to make it more accessible for the general public. I am so thankful to be welcomed with open arms by John Burns, my mentor, and the rest of the Mega team. They gave me so many incredible opportunities this summer, such as being able to participate in outreach events, attending NOAA photogrammetry trainings they hosted, and most importantly, jumping in on one of their projects this summer, namely the Opihi project. Now, when I started the summer out, I had a very vague idea of what an Opihi was. It's just this silly little guy. But over this summer, they have become close friends of mine, as I have spent all summer looking for them and counting them. They now hold a very special place in my heart. But in general, this endemic limpet to Hawaii is known as a traditional food source, very popular, although I've never tried it. And its shells can be used as tools and jewelry alike. They have been connecting people to the ocean and their ancestors for a long time. The Opihi project aims to assess the health, productivity, and resilience of the rocky intertidal habit that they call home, and how it changes in relation to sea level rise. A big part of our project is developing ways to make this assessment accessible for the public, finding ways to monitor an entire coastline easily and efficiently. Now, real quick, let's go over the hypotheses that we're looking at here, of which we have two. The first being that we expect distribution of Opihi to expand in the winter and contract during the summer due to seasonal changes in water level and food availability. And secondly, that sea level rise will change the abundance and distribution of Opihi in coastal areas. Now, you might notice that one of these is seasonal, and I was only here for this summer, which means this project definitely outlives my internship and will continue into the summer of 2026. The surveys I was a part of more so built the bricks for future surveys. So we were figuring out the methods and kind of how we want to proceed with the project in the future. Now I'm going to show off where we were all summer. What was I even doing? I was over at Kalae Cultural Reserve. If you haven't heard of it, it is a 3.6 stretch of coast that has been under active stewardship for around a decade now. It is a coastal area within the Apua'a Kaupulehu, known as a traditional fishing spot with cultural significance, such as Papakai and Limu, most importantly, Opihi. This area is so special and it's just gorgeous. It's up on Kona side and it will be open in the summer of 2026. Special thank you to our kapuna and guide, Auntie Lei, who has been overseeing
Kalae for a while and has given us permission to do our surveys here. She has been such a huge help, and I just really want to make sure she gets her dues. Also, if you ever visit, go talk to her. She's a lovely individual. Now, drives up here every day were brutal, I will say, and walking along the coast, although it is beautiful, is brutal as well. I can attest to this myself. My balance and my ankles have definitely grown stronger climbing up and down the coast every single day because it is mostly lava rock that is sometimes a little unstable. So if you do go, definitely wear your felt sole toppies, please, for your own safety. Now, with a spot like this, it is not only hard to navigate on foot, but probably even harder to keep track of with the tides rising and just everything changing so often. So the way we decided to approach this was through photogrammetry. Now I want to talk about our main star here, the main character in my story, the GoPro. I definitely do not have a bias towards GoPro cameras. However, they are my favorite. I'll get to how we use them in a little bit, but first I just want to go over a couple of the features that the GoPro model we used over the summer has. It takes super high resolution photos and everything is just super clear. It has this very wide lens, so every picture we take captures a lot of area. That will be very important later. It has a time-lapse setting, so you don't have to like click the button every couple seconds. It just kind of goes for a little bit, and that is super helpful when we're going through our transects. It also has this this wasn't really relevant to our project, but it has a bunch of little screens and it's super user-friendly. So not only does it have a screen on the front, but a screen on the back that you can just scroll through at your leisure. You can check your photos in the field, which I know doesn't sound like a big deal, but when you can check the quality of your data in field, you can figure out whether or not you need to retake it right away, which saves us a ton of time. So it is my favorite camera that we have used in the lab so far, and definitely the one I am the most comfortable with. So yay, GoPros. Now on to the actual photogrammetry part. We basically are creating structure from motion, or SFM models, of the transects we set up along Kalae. Basically, we take a bunch of photos and stitch them together. For this to work, you do need 70% overlap between all of your photos, which is super important so the computer can actually line them up to stitch them together. And we take extra steps to ensure this, such as using a lawnmower pattern when we are transversing our transects, like our little diver is here. You kind of start in one corner and you zigzag across. It's a very fun process, I will say. And our routine was to find a spot for a transect, measure it out, take our photos, and then walk around 100 meters down the coastline and set up another transect. We had around 20 transects in total, so we got basically a lot of photos, a lot of data, and we were able to make 20 beautiful, beautiful models, one of which I have a little GIF of to show you guys. So the models we build end up looking like this. It is pretty much a real life, it's a photorealistic, obviously, snapshot of this area in time. Sorry if it's glitching a little bit, but basically we can get so much information from models like these. I mean, you can see the elevation right there. So later on, we can see how the tide affects it. You can see all of the different colors and crevices. You can see the Limu and Opihi using these models. It really is amazing what we can accomplish with these. And that's just looking at this. We can also quantify all of these observations we are making through ArcGIS, which ends up looking like this. So we use the digital elevation model and orthomosaic, crop it down to the size we need, and we end up with this nice little map. The one on the top left is not really annotated, and then the one on the bottom is annotated. So we are looking at the different zones where Opihi are going to be. We have them categorized in 3 different ways: the black zone, the mixed zone, and the pink zone. Starting with the pink zone, that is where you're going to have the most productivity, the most CCA, crustose coralline algae. Lecture before, we do have that later. That's the time to see. So That's where most productivity is going to be. And it is kind of pink. The black zone is basically the opposite. It's super high and dry, and the mixed is just everything in between. And we also count all of our OPE individually, which is all the white dots. They didn't show up as well as I would like, but I hope you can see them. There are, I think, around 136 on this model. And we ended up counting a total of 2,919 OPE across all 20 models. So a lot of OPE, yay! Everything is scaled as well, so we can measure the OPE using these models, which is super helpful because counting them in field, as much fun as it is very difficult and takes a lot of focus that I personally do not have. So I prefer this method many times over. Now, what we can get from stuff like this are just generalized statistics, and a couple of which I have just extracted to share with you all. I'd like to talk about the prime OPE real estate that we have here. OPE do have preferences. They are very specific little guys. They seem to really prefer the pink zone. That is where they grow big and strong. That is where our largest OPE are going to be, right there at the shoreline. They are adventure seekers. They really like to hang off of cliffs where all of the waves are splashing them constantly. I wouldn't think that's ideal growing conditions, but the OPE seem to love it. Black zone is kind of the opposite. You're going to see a lot of smaller individuals, but they are still going to be there. And right here, I have a figure showing OPE density by zone with zone at the bottom and OPE per meter squared on the side. Pink and mixed actually had a very similar density, but mixed has way more variability. It is a wild card, if you will, kind of just everything, everywhere, mixed zone, you really don't know what's going to happen. But yeah, no, we can just look at things like this, and when the seasons change, we can see how these areas change as well, and how that affects maybe where the OPE want to be. And moving forward, obviously, touching on the seasonal things again, I will not be here for a lot of the seasonal developments, but I am very excited to see how things will change, how the sea level rise will affect where the zones are, and everything like that. I didn't get to cover a lot of the drone stuff we did. Thank you to Kainalu Stewart for helping us out with a lot of it. Unfortunately, most of it flew right over my head. It helps us with mapping, and it looks amazing. But that might have to wait for another day. We can keep moving forward and connect this research to our communities, helping them understand data like this so they can monitor it as well, because it's going to take a lot more than just us. I would like to give a special thank you to everyone on screen, including PyCask, because there are so many people to thank, and I am beyond grateful for this experience. It has been a wonderful summer, and I look forward to getting more opportunities like this to connect with people and the environment around us. Thank you.
We have a couple minutes to have some questions if anybody's got any. And that includes people online too. If there are any questions online, just unmute. Jordan. Oh, waiting for a mic.
I was just gonna ask about your process of counting them. Did you tag them a specific way so you didn't count them twice, or how did you go about that so you wouldn't double tap it?
Yeah, it's very easy to double count OPE when you're there in person, but thankfully the photos we have are just static, so you can kind of plop a point on it and move on to the next one. I'm sorry, I don't really have any zoomed in photos, but yeah, no, counting them in person, I will be honest, I don't know if I recount them a couple times. We did that, I think, once with some other people, and it was very difficult, but online we can just kind of put a point on it and it is counted, which is very helpful.
Cool, thank you.
Of course, thank you.
Okay, well, let's move on then.
Thank you.
Can I have the mic?
Yes, you can.
I can figure out how to get it off.
Yeah, there you go. All right, moving on. So Kyoko, you're up. So Kyoko is a rising senior in the Global Environmental Studies program here at UH Manoa. This summer, she worked in Elizabeth Maiden's lab with Jake Linsky, learning how to improve whale identification by AI in satellite imagery. Very high tech. Over to you.
Thank you. Hello, hi everyone. My name is Kyoko Suzuki, and today I'm here to talk about whale population estimates using satellite data, particularly how to enhance the accuracy of satellite imagery population estimates.
Was that supposed to be a movie?
Is it video or?
Apparently not.
Oh, okay.
Sorry,
That's okay.
didn't translate.
Well, this was a video.
Imagine the whale swimming.
Of humpback whales migrating to Maui. Every winter around January to March, they migrate here all the way from Alaska to breed, give birth, and take care of calves. Warm and shallow water here in Hawaii protects them from predators and strong current, and it provides them with breeding areas. So here's what's happening with whale population globally. The whale populations are now facing new threats from climate change, vessel overlaps, and food availability. However, overall, there's a rebound in many whale species after large-scale whaling stopped. This is due to reduced direct hunting pressure and long-lived whales that led to population recovery. What about population here in Hawaii? The population reached highest in the 1990s and 2000s. However, whale populations have been declining since the mid-2010s. Sharp declines in North Pacific whales. There's a sharp decline in North Pacific humpback whales, reproductive success, and calf survivals. In fact, their visit to Hawaii declined by 34% between 2013 to 2018. This is due to heat waves caused by El Nino, North Pacific oscillation, possibly climate change that reduced primary production and prey availability. Whales play a vital role in maintaining healthy ecosystem. For example, their horizontal migrations carries nutrients from nutrient-rich area to nutrient-poor area, and their fecal dispersion also promotes plankton. So it's important to conserve their population. In order to conserve them, accurate population estimates are important. There are so many ways to measure whale populations that include boat survey, acoustic monitoring, photo ID, and drones. Personally, boat survey and photo IDs are one of my favorite surveys, but unfortunately, they're very time consuming and they need somebody to do the survey. So our solution is to use satellite images, which allows remote access and which also has a potential for automatic detection, which can be cost effective too. You might be wondering what population estimates on satellite images is. So this is what it looks like. It's very similar to Google Earth, but instead of looking for like houses or buildings, you're looking for whales in the ocean. It's a very high resolution satellite images, and we're using the ones that were taken in Maui. It is great for monitoring marine life, for population estimates and behavioral observations, and it has a potential for AI detections, which can be quick at processing large data. Here are some examples of what whales look like. You can see there's 3 whales there. There's another one here. And on this imagery, we were able to find 11 competitive whale group. it is possible that there's one female right there being chased over by 10 other male whales. Here are some more details on what whales look like on satellite images. And what I want to emphasize here is that they all don't look equal in terms of certainty. For example, and therefore we categorize them based on certainty on a scale of 0 to 2. Those 0 whales are the ones that you can kind of see that there's something, but you can't really tell that these are whales. 1 whales are the ones that you can clearly see the shape, but they're not clear enough to do do species identification. And those 2 ones, you can see long flippers over there, and you can see that they're humpback whales. At the same time, satellite images are not perfect either. It's not always clear. Oftentimes they're covered with clouds, glare, and wind and waves, which makes it difficult to detect whales. And these make me wonder if there's actually an accurate estimate in whale population estimates using satellite images. So here are the 3 key elements for accurate population estimates. First of all, you need a count. And second, you need availability. And third, detectability. Whale availability is the likelihood the whales are on the surface water that are visible from satellite images. This is a dive profile of humpback whales. And as you can see, they're not always swimming at the same depth at a different time. And satellite can only detect whales at a certain depth. Whale detectability is the likelihood that any environmental conditions prevent satellite from capturing whales. That includes clouds, glare, and wind and waves. So the aim of my project was to improve the accuracy of whale population estimates using satellite imagery. And to do this, the goal was to determine what factors influence whale availability and detectability and how. I listed 5 variables that could affect detectability and availability of whales: depth, clouds, wind waves, glare, and turbidity. And for this project, I decided to use 2 variables, depth and glare. I used depth to compare with whale availability and glare with whale detectability. For depth, my question was, how does depth affect whale availability on satellite imagery? My hypothesis was that there are more whales present in shallower water as they're more likely to be on the surface. Here are the methods that I used. I started off by annotating whales on 42 satellite images on ArcGIS. I categorized whales on a scale of 0 to 2 based on certainty, looked at the distribution, and finally analyzed whale and depth relationships. Here are the results that I got. This shows the bathymetric map this is a bathymetric map with a color gradient highlighting depth variables. And I plotted whale sighting over a bathymetric map. And this is the plot of whale sighting by depth. And as you can see, there are more whales at a shallower water than deeper water, which could be caused by two causes. One is whales are in shallower water. Whales in shallower water are more likely to be in the surface, and therefore more detectable in satellite imagery. And if this is the case, we need to consider bathymetric data when doing the population estimates. And the other is whales actually prefer shallower water for their habitats, and therefore there are fewer whales in deeper water, and which supports a lot of studies that show that mother and calf actually prefer shallower water. For glare, my question was, what time of the day and sun glare produce glare? and affect whale detectability. And my hypothesis was close to sunrise or sunset when the sun is low. I started from recording glare and comparing detectability and glare relationship. I took latitude, longitude, time, and glare level of a specific grid, and finally analyzed what time of the day and sun angle produce glare. This is the results that I got from comparing glare and certain whale sighting. You can see that there are fewer 1 and 2 whales as glare increases. You can also see that there's about equal number of 0 whale sightings, which indicate that annotation mistakes increases as glare increases. I also found out that glare was the strongest at 2 p.m. and at 45 angles, which may correspond to a sun angle at 2 p.m. in Maui in winter. But this actually was not my original hypothesis. And I realized that we had a lot of data that was captured in 2 p.m. So in order to determine this, get an accurate result we need more systematic approach. Here's my conclusion. The aim of this project was to improve the accuracy of whale population estimates on satellite imagery. And the goal was to clarify what affects availability and detectability on satellite imagery. My objective 1 was to examine depth at a variable that affects whale availability. Objective 2 was to examine glare at as a variable that affects whale detectability. And my future goal is to expand more different variables and do more annotations for training AI. I would like to thank Dr. Jacob Linsky for mentoring me throughout this project, and Makoto Murata for providing me with more annotation data, and Elizabeth Madden's lab and marine mammal research projects for this support, and SIP program funded by Hawaiʻi Sea Grant and PICAS for giving me this summer research opportunity. Thank you.
We have time for a question or two. Anybody have anything? Sara, you're so good for picking questions.
That was super cool. So I'm wondering if, because I'm not used to working with satellite data and particularly like detecting whales. I don't know how you could tell those blobs were whales. That's amazing. But I was wondering if there's anything that you folks have done that allow you to validate the satellite with like in-person field surveys. So are there any satellite images that come from days when folks have been out on boats? You can kind of like use that same data to figure out if you're getting it right.
Not the one that I annotated for this project, but we were just talking about it with my mentor yesterday that we found 4 objects that looked like whales, but we weren't really sure. And we also we knew that there's a data from Pacific Whale Foundation. Oh, cool. So we could possibly use that data to see if those are whales. Oh, and what's really cool about it is that on satellite imagery, a lot of times you can see whale watching boats or whale survey boats really right next to or really close to whales. So cool. If there are multiple whales, it's possible that there's also survey going on. So
that's cool. Yeah, so you can use other markers to find them too. Nice. Thank you.
Okay. All right. Well, let's move on then. Thank you. All right. Mike. Thank you. All right. Moving on to our next techie talk. Cienna is a rising junior doing a natural science engineering degree at Kapiolani Community College in UH Manoa. For this summer project, she worked with Troy Heitman at HNEI on modeling environmental impacts of wave energy installations. So, find a place to put that if you.
Yeah, I think over here.
Oh, no, hold on. Yeah.
Okay,
Okay, that'll work. That'll work.
Aloha, everyone. This summer, I worked on simulating wave energy converter installations in Hawaii for sustainable energy advancement. It is a SWASH-based numerical modeling study, and SWASH stands for Simulating Waves Till Shore. Around the world, we're seeing a rise in electricity demand by 2.1% per year. And in Hawaii, we have a very unique situation where we are on an island. So our electricity is mostly dependent on petroleum, and it's the highest in the US. And by state law now, we're actually required or trying to be 100% renewable electricity generation by 2045. And now we are currently at only 33%. So in order to obtain that goal, we are going to have a diverse portfolio in sustainable and renewable energies. Currently, we're relying on things like solar and wind and a small part of ocean energy, which is geothermal. But in Hawaii, we have yet to utilize wave energy in our systems. And globally, there is an abundance of wave energy. But specifically for Hawaii, we do have a unique opportunity in a sense where we are placed in an area where we have constant and reliable wave energy, with some wave energy flux exceeding 30 kilowatts per meter. And in this bar graph here, we have a comparison of our annual energy demand versus annual wave energy around the islands. The bar graph bars with the numbers on them are different contour depths, and then the black is for total electricity demand in 1990, and the blue is 2015. So really what this graph tells us is that if we are able to harness all the wave energy around all the islands, we would more than exceed our total demand. Of course, that's totally infeasible to harness all energy around the islands, but it's really good for showing the scale of energy that we're really missing out on. So in order to harness wave energy, you do have to use a wave energy converter. So for our studies, there are actually many wave energy converters being studied here in Hawaii, but we specifically focused on oscillating surge wave energy converters, which is stands for OSWEC. But this device here is called the HAWSEC, which stands for Hawaii Wave Surge Energy Converter. And essentially, it's a flapping device. So as the waves pass through it, it will push and pull the flapper, and it's connected to a piston and connected to turbines and a PTO system, which then generates electricity. But essentially, this item is fixed in the seabed in shallow near shore waters. This particular wave energy converter has been gone through multiple testings, including one at Oregon State University. And that data set is actually what we use to validate our SWASH model. Though you would typically not just deploy one wave energy converter, you would typically deploy an array of wave energy converters like you see on the far right there. And
wave see the good uncle and watch the whole thing.
And where was that supposed to happen? Or
it's in Hawaii.
Okay. Yeah. Typically you would do an array, but there's benefits to doing an array like this. And it's not only energy generation, but you can also use these as coastal infrastructure protection. As you can see, it does protect some of the coastline if you do them in arrays. On the left, there are two different models other than SWASH presented here, both of which are not as detailed as the SWASH modeling system. So that's why we chose the SWASH modeling system, because it's
Although both of these systems are good, they do not provide the same, there we go. The same amount of detail as SWASH is able to provide. SWASH is able to resolve areas around objects and around the shoreline as well. So, what happened there? So my project objective was to really utilize the OSU wave basin data to validate the SWASH modeling system and evaluate its interactions with nearshore environments and assess its energy extraction and potential shoreline impacts. So to do so, first is initialization. So basically I take the bathymetry of the OSU wave basin, which is basically just a flat basin with a slope, like a slight slope there. And I'm able to put that into SWASH and replicate the wave that was actually generated at the basin so that we were able to accurately model what was going on. And then next is convergence. So convergence testing is essentially a way of checking whether increasing the resolution of numerical model changes the results in a meaningful way. So if the results don't change beyond a certain point, then the models have said to converge. In SWASH, the water column is divided into several vertical layers that move along with the surface. So for this study, I ran 1, 2, 3, and 4 vertical layers, as you can see in the testing at wave gauge 3. And we're able to see that essentially it converges at 2 to 3 vertical layers. And anything after that, it's not worth the computational excess. And then next, we did validation. So because they did test at OSU already, we did have wave gauge data at these points on the left here. And that graph came out extremely light on this. But basically, it compares the experimental data to the model data. And when you remove the phase offset, it actually lines up fairly well. You can see on the histogram there, I'm able to quantify the amplitude without any of the time variances. And then we're able to run some aerometrics to compare. Sorry. So I was able to run root mean square error, which gives you like an overall sense of accuracy relative to the observed data. Mean absolute error, which is the average of absolute differences without any outlier effects. And then the bias. So the bias will tell you whether the model systematically overpredicts or underpredicts. And for wave gauge 6 is on the bottom right over there. Oh, hold on. There we go. Okay, so this is the summary of error. As you can see, overall, the error is a whole order below the actual wave height. So that being said, we were able to validate the model against the physical data. And so it shows a strong potential for simulating real-world wave energy converter performance. And lastly, implementation. So now that we validated the basin, now I can put things in the basin. So first we started off with a stationary OSWEC, and basically it's an oversimplified version of what a wave energy converter looks like, and it's just a block in the water. But this is essentially so that we can suss out any variances or anomalies that might occur before adding any more complex motions or blocks in the water. Unfortunately, I did try to implement a pitching or a flapping wave energy converter in there, and there were some challenges along with that. So instead, we moved on to open ocean.
So this is Kilauea Observatory, which is around Kewalo Basin. And so what this really shows is that we are able to input more complex bathymetries and different wave parameters within the model, and it shows up quite accurately what would actually happen. So yes. So really, in summary, we were able to show that using the wave basin data, we're able to accurately model what would occur in the real world, essentially. And for future work, it would definitely need to include a pitching WEC, and then implement that into areas like Kilauea Basin or Observatory and other places around the island. Thank you. Thank you to my mentor.
Yeah. Very cool. Anybody have any questions? On mine as well? Brad's got one. Oh, I need that. Sorry.
Interesting presentation. Thank you. I'm wondering in general, and I don't know if you looked at this, but how much does wave period matter? I'm wondering, like, comparison to like a leeward shore, like South Shore, that last model you showed is going to be more long period south swell compared to like a windward shore that might have a lot more shorter period and mixed swell.
Mm-hmm.
Did you look at those differences? Is that going to change the performance of some of these devices? Do they need more regular?
Unfortunately, I that would be saved for future work because I wasn't able to implement any of the actual moving WECs, so I won't be able to see what the effects are quite yet. But that is something that has been discussed with my mentor.
Okay, thanks. It looked like the testing was though in like a more regular oscillating wave. Yeah.
We are able to, so on the real world though, we are able to implement the various wave conditions, and so we would tailor them to the areas that we are there in. So the model is able to handle that. Mm-hmm. It's just I did not get through it. Yes.
Great job. Thank you.
So I have a quick question. So are you going to be looking at this in terms of, I mean, you had talked about what the effects are on the environment from putting these things in, but is it also in some ways to determine where would be the best places to put them? Like, can you use that too?
Yeah, so when you are testing out the WEC arrays, we would also test the output of energy as well. And we would essentially run a comparison of all the areas. I believe he has some testing areas in mind already, namely on, what is that? East side?
Windward?
Yeah, okay. But east side and up into Kaneohe, there's some areas of testing that they have in mind. Yes.
Okay, cool.
Oh, sorry.
One more? Oh, wait, we need the mic.
Oh.
Thanks. First of all, say I love the chaos of seeing all the interactions once it hits the shallow water in the reef. But I was curious, what kind of like benthic resolutions does this require? At what point will it start to break down as you kind of downsample the input data?
Are you asking about the resolution of the satellite imagery in order to get
like kind of like the benthic inputs and everything?
Yeah. Thankfully near islands, well, near any islands, we have a pretty high resolution of satellite imagery. For places like Planet Lab, which is like a specific site for satellite imagery, their resolution is about 3 meters per pixel, which is fairly reasonable. But as you get further out from the island, the bathymetry matters less than near shore. Yes, it matters, but not really in our case, because we should have a consistent data set because we're so close to shore. I believe for where I got this, which was Google Earth, I believe theirs is 10 meters. So it could be.
Okay, I guess we will call that.
I hope that answered your question. Sorry.
So we have 4 more talks. So I have built in another brief break so that people can get up, go to the bathroom, eat a cookie, whatever. So let's just make this one 5 minutes. Is that okay? Okay, so we will start back again at 2:45 with Kalae. Fly, be free. So beginning our next chunk of talks, our last chunk of talks, which are more focused on kind of broader ecological, environmental sort of questions. Kalae is another rising senior majoring in both biology and political science at the University of Portland. She worked with Sara Kahanamoku-Meyer on recovery of atoll ecosystems after human and natural impacts in Papahanaumokuakea. Enlighten us. Tell us all about it.
Great. Okay.
Did you want to use the remote? Or you could just clicking?
Yeah, I can just click. Okay. Hi, everyone. My name is Kalae, and this summer I worked on characterizing the geo-ecological resilience of atoll islands. Sorry. Okay, so I wanted to start with this Olelo no Eau, which is He Puko'a Kani Aina. And this directly translates to a coral reef that grows into an island. This concept is super important when we talk about atoll reef islands, as they are composed entirely of reef-derived material. So wind, waves, organisms break down the offshore reef, and then that sediment is then transported to the islands to grow. For our summer project, we focus specifically on Papahanaumokuakea Marine National Monument, which is located about 500 miles northwest of Oahu. And this place is super important culturally, as these islands are thought to be the ancestral or kupuna islands. So it's super important to protect these. And we focus specifically on Lalo Atoll or Tern and East Island. Those are located within there. So not only is this place culturally important, but it's also super important ecologically. Lalo is a nesting site for about 90% of honu, or our Hawaiian green sea turtle, and 20% of the Hawaiian monk seal. It's also sensitive and critical habitat for millions of birds and a bunch of other really cool species. Lalo was also in the direct path of a hurricane in 2018. It was a Category 3 hurricane with sustained winds of up to 150 knots. So it caused detrimental impacts to this atoll. On the right there is a picture of the reef. On top is before the hurricane, and on the bottom is after. So you can see that there is an entire loss of aggregate carbonate habitat. And then this is a picture of East Island or Hikina Island on the left and Tern or Ewa Ewa on the right. You can see for East, it lost about 95% of its original landmass, which translates to about 66,000 cubic meters of sediment in a single day. So you can see there's a small little island here that remained. And then Tern Island was modified during World War II, so it was hardened and dredged, as well as there was an addition of a seawall. So it ended up gaining 40,000 cubic meters of sediment. 5 years later, however, Tern Island looks pretty much the same, and East Island has recovered to about 56% of its original landmass. So this led us to a research question for this summer about how are low-lying islands able to recover and maintain resilience after major disturbances. To do this, there were 2 research cruises, one in 2018 prior to the hurricane and one in 2021, 3 years after, that collected sediment samples from various locations on these 2 islands. The sand was then sifted into different size fractions, and then because it's just sand and we can't really tell what's in it, it was sliced into thin sections so we could identify the components. So on the picture on the right, this is a sample from Tern Island, and you can see there's a little piece of rust from the degrading seawall. And then on top is CCA, which is important and I will get to later. So after identifying each of the samples' composition, we used a PCA or a principal component analysis. It doesn't have the, oh, the ellipses are a little light. But on the left, so this graph groups together samples based on how similarly they are to each other. So the pre-hurricane island samples, which are more towards the left, are grouping together with a higher composition of CCA or coralline algae. And on the right is the post-hurricane and offshore samples that are grouping together with less CCA and more coral. So this kind of started to tell this story of selectivity of sediment source. So prior to the hurricane, it was a lot of CCA getting tossed onto the island. And then after the hurricane, we can infer that the offshore sediment source or offshore sediment was basically picked up and dumped onto the island. And then, we also used Tern Island as a way to further prove this idea. Because it was hardened, it created this nice 45 centimeter layer of sediment deposit. And then using this histogram, we were able to compare the composition of the hurricane deposit to the offshore and found that it was pretty similar. I think I updated this histogram later. But yes, so it's pretty similar to the offshore source. And then what's important to note here is the relative abundance of CCA or coralline algae. And we found that there was significantly more CCA during our normal intervals rather than during a disturbance. So this led us to ask the question about functionally what is happening in the reef during this initial period of recovery. So crustose coralline algae, or CCA, is super important in helping reefs recover. So through research, we found that it is found basically everywhere. And after disturbances, it acts as a reef binder. So it glues the coral rubble together to kind of form a more stable environment. It also sends out chemical signals into the ocean to promote coral larval attachment and has species-specific preferences. So super important in our reef ecosystems. And then this picture is from looking at one of our thin sections. So this is what CCA looks like under the microscope. So this is before, and then you zoom in and you can see this. So we identified these small circles as the reproductive structure, which is also called the conceptacles. And through measuring the size and density of each conceptacle, we actually found that it shows a classic life history trade-off. So prior to the hurricane, there was less conceptacles over the area. So this linear regression on the right has mean conceptacle area versus conceptacle density. So basically showing that there were more conceptacles after the hurricane over the area. So showing that the CCA reproduction rather than growth. So this suggests that after a big disturbance, the CCA shifted its energy toward reproduction, possibly as a strategy to quickly repopulate and recover. All right, so understanding reef recovery is crucial, especially as climate change brings stronger and more frequent storms along with ocean acidification, all of which makes reefs more vulnerable to damage. So we've already seen the consequences. This picture on the bottom left here is of Tern Island after a storm in 1953 that had already begun to damage the seawall. And then on the right is now the seawall continues to serve as a barrier for these this marine life. And also there's PCBs being released or chemicals being released into the ocean and on land. This photo here is the low-lying islands within the Pacific. It's home to more than 1 million people and has really biodiverse ecosystems. So it's important to understand what to expect in the future and how to possibly protect them. Okay, so for my research, this kind of led into the idea of how to use this science to inform our management decisions. So how do we manage atoll reef islands for resilience? Further research should definitely look into kind of the CCA ecology. So understanding what its role is in reef recovery. So how it can serve as this primary successor. Also understanding long-term dynamics. So looking at past natural experiments of climate change and identifying what to predict and how to prevent this. And then finally, going back to the Olelo No'eau in the beginning, He Puko'o Kani Aina, looking at now that we understand that the sediment sources that are feeding these islands come from the offshore, it's important to protect this source. And then the Papahanaumokuakea Marine Management Board has talked about working on restoration to these islands as well as protection. And this idea of lalo I lalo, which means lalo returns to the sea, kind of deals with the idea of human intervention in these sort of natural experiments. So when to let these islands be and when to step in to preserve them. So lots to think about, but this summer, just understanding that CCA plays a huge role in reef recovery provides a small stepping stone into what to look for in the future. And then I would like to thank my mentor and all the people in the Mega Lab and Mano Lab for everything they've done and this amazing experience. Thank you.
All right, we have time for a question or two. Anybody in the room or online have a question?
Yes.
So with CCA being like a super important like recovery thing for a lot of coral reefs undergoing like bleaching and that kind of thing, do you think it would help if we were to put more CCAs into struggling reefs, or how do you think they could be used?
Yeah, so that's kind of something that I was super interested in when we first saw CCA's importance. So through extensive research and reading papers, we found that CCA actually can withstand certain conditions better than coral. So like with ocean acidification and rising temperatures, I think that could be definitely a really good next step to look for research. Yeah. It hasn't been done yet to my knowledge, but yeah.
Okay. Any other questions? Really nice work. Cool. Thank you.
Thank you.
All right, up next is Jordan. So Jordan is also going to be a senior majoring in environmental studies at Hawaii Pacific University. There you go. She's been working with Carmela Visa from HPU, looking at fish communities across different habitats on the North Shore of Oahu. Go ahead, Jordan.
All right, hello again. My name is Jordan. My summer project really focused on linking the different fish community patterns with habitat conditions at Waiale'e on the North Shore of Oahu at those 3 different sites. So why this study matters as a whole, Waiale'e is a culturally and ecologically significant site in the North Shore. It's currently undergoing monthly restorations with hyacinth removal and the Lo'i revitalization. And it's always been known for its historic aquaculture and its agriculture. So monitoring these different fish communities actually helps track the impacts of restoration on the species and habitat health as a whole. So historically, these maps are from the 1800s and they show what the land used to be used for. You could see that little kind of sideways L-shaped pond. That is what the local vai is throughout this presentation. That has always historically been used for aquaculture, so different fishing and stuff like that. And then the lo'i and waiale'e, you can kind of see, I'll show it better in the next slide, but that was used for taro patches and then just dry land agriculture, which was key to traditional Hawaiian food systems. So this is an overhead view. You can see waiale'e is within the green circle, and it's kind of like an open water area mixed with the freshwater hyacinth. And then the brown circle, that's what the lokolo'i is. There's also kind of like a open water area. It's a lot smaller current day than what it's showing on the Google Maps. And then we did a transect through that freshwater hyacinth. There's more open water now as well current day. And then the blue circle, that's the lokovai on the other side of the lo'i. So this is just another closer look. I'm sorry, but I just think like the look of these sites is very important to the study as well. So waiale'e, you can see the open water with the kuana next to it, which is separating the land from the open water. And then you can see the fresh hyacinth on, or the freshwater hyacinth on the horizon. And then the lokolo'i, you can see the kalo that's growing in now. Very excited about that in the shallower area. And then there's an overhead picture where you could see just how much freshwater hyacinth is. That purple flower is actually a flowering freshwater hyacinth. So you could see like we did it in the open water, and then also in that hyacinth, we laid the traps and everything as well. And then the lokovai you can see is completely hyacinth free. It's just an open water fish pond. So my main objective and hypothesis was I wanted to look at the different fish community composition to see what fish we could find in the different habitats, kind of based on their vegetation and their water quality levels. My hypothesis was that the lokovai would have the most diverse community just because it is that open water fish pond, and that the lokolo'i will have the least due to all the hyacinth coverage that we kind of had to walk through and maneuver through. And that waiale'e would be intermediate, but impacted by the low dissolved oxygen that we can see if you've been to a work day. We actually disturb all the water and kick all the mud up. So the main method we did was fish capturing. We had these big fish Lollapalooza days. They were 2-day surveys per site. We used 17 baited traps. We had 10 fan traps, which is that kind of geometric one. And then we had 7 minnow traps, which is a cylindrical one. Fish were measured, sexed, and tagged with visible implant elastomers, which is actually that orange dye you can see there. We injected it kind of like near its eye. I don't know if you can see it, but it's like a little neon dye, so we can kind of like log those fish. We used dough balls, which is at the bottom. They kind of look like weird snowballs, and they're made out of peanut butter and bread. And then we also just used shrimp. And then for the water quality, we took the water quality with the YSI at the top right corner by just basically putting the probe in at the top level of the water and the bottom level, and we got the dissolved oxygen, salinity, temperature. But then, yeah, we also put the fish in aerated buckets after, which is like recovery for them, and then we release them back into their locations. So total fish captured, we captured a total of 1,162 fish across the 3 sites during just the month of July. So this whole research is just giving us a snapshot of data. We can't really like assess the data without a timeline. So this is all just about July. By taking out the fish that were smaller than 4 centimeters, because you can see that really skews the data, you can see that the lokovai actually had the most diversity among the sites, and it also had the only native fish found in all 3 sites. So the lokovai had Oupua kupa as well as Oupua neniha, and it also had catfish and tilapia, whereas the other 2 sites just had tilapia and the catfish along with the fish that were smaller than 4 centimeters. So specifically about the fish that were smaller than 4 centimeters, a lot of them were fish like mosquito fish, which you can see at the top. That's actually not native to Hawaii. There were 662 found at waiale'e, and 319 found at the lo'i, and none found at lokovai, which was very interesting. That could possibly show active reproduction, meaning that these fish are being reproduced, and it may actually limit the native fish in waiale'e and the lo'i. So with that, wanted to look at specific fish to see how the habitats were affecting the size distribution. So not only just the biodiversity. So we compared different tilapia sizes, and we saw that the largest average size was found in the locovii, which kind of supports the hypothesis that because it's a freshwater spring-fed pond, that it had like the most ideal qualities for the fish to thrive in. So the largest size were found there. The lo'i had the intermediate size, and waialae had the smallest size tilapia at 7.5 centimeters. So again, it does kind of, with the size distribution, you can see how it's showing that habitat quality could affect the growth of specific fish. For catfish, we only collected a few, so there wasn't enough data to do a box plot, but a general bar graph showed that the biggest size was found at the lo'i, and then the intermediate size was found at the locovii, with the smallest size again being found at waialae. And this is because there's, again, like that open water bank that's right at the start of the lo'i by the locovii side. And because locovii spring fed, some of that fresh water gets into the lo'i. So maybe that was attracting the catfish to that area, because all the catfish that we caught in that area was by hook and line sampling. They weren't found in the traps to my knowledge. So that could be because of the prey availability in that area, because of the small fish, and then also because of like the habitat qualities itself, with like all the places to hide in that habitat. So with the water quality, again, we took it with the YSI, which is in the corner. We found that the salinity differences were very minimal across all 3 sites. So the sites were extremely fresh compared to ocean water. Ocean water is like above 30 parts per thousand. Every site was less than 1. So it's very unlikely to be a major driver of any differences that we find throughout the research. So we really just honed in on the dissolved oxygen and the temperature. And we saw that waialae had the lowest dissolved oxygen of the 3 sites and the highest temp. And then the lo'i had moderate DO with moderate temperatures, and the locovii had high dissolved oxygen and then also moderate temperatures. But you can see that there's like the lo'i and the waialae location have more variability than the locovii does. And for the lo'i, that could be because it's almost like 2 sites in one, with the freshwater hyacinth and then the open water area. And then waialae, there could be like a multitude of reasons for that. But you can see again that the locovii has the ideal conditions. So what does this really tell us? Waialae's conditions in comparison to the 2 other locations kind of aligns with dominance in the tolerant fish like tilapia. Tilapia can really thrive in harsher conditions than native fish can. So that's why we found, or why we think we found, tilapia species and fish that were less than 4 centimeters. The lo'i had moderate values that supported both fish less than 4 centimeters and tilapia, but its physical factors like the freshwater hyacinth could really limit the diversity and impact the variability that we found. And the native fish were, again, only found in the locovii, which had the highest dissolved oxygen and no hyacinth vegetation found there at all. So it shows that the water quality strongly does influence which species can actually thrive at each site. Higher water quality in this situation with higher dissolved oxygen and low variability are linked in this research to greater fish presence. And then the top fish is an Oopu akupa, and the bottom one is an Oopu niniha, which were both found in the locovii. So overall, sites with the higher dissolved oxygen, like the locovii, supported the greatest biodiversity, including the native fish, Oopu, and it also had the largest tilapia. Warmer and more variable conditions, like waialae, had the smallest tilapia and smallest catfish, and was dominated by native fish that were less than 4 centimeters, which shows that those conditions could potentially limit growth and survival of larger species. And catfish were the largest in the lo'i with the hook and line sampling, potentially due to abundant cover and food sources and maybe the fresh water coming in from the locovii. And overall, the water quality and habitat characteristics appear to directly influence the fish community composition, as well as the species presence and the size distribution across all the sites. So what's next? Continue monthly sampling to look for seasonal patterns in water quality in fish communities. Because again, this was just a snapshot of data, so we can't really determine a lot from it. It's just what you see is kind of what you get with it. So to collect water data at individual trap locations too, because we're doing it at site level. But I think that if we were to do it at trap level, then we could see like better, like, oh, this specific fish had this specific water quality habitat. So it wasn't just level, because different places in the site could have different water quality. And then we would compare seasonal trends to restoration efforts. So before and after snapshots to track progress and guide future management, like doing a fish day before restoration day to see how the restoration day and then after to see how that impacts the water quality. And then I just want to say thank you to the University of Hawaiʻi Sea Grant College Program for giving me this opportunity, and the University of Hawaii and North Shore Community Land Trust for being fantastic community partners, and Dr. Vitsa, my mentor, and my fish team.
Very cool. So that sounds promising that the fish pond actually is good for local fish.
Yes. It was really
Nice.
fun to see all the native fish that we caught.
Yeah. So if we put in new ones, if we fed with more fish, do you think they'd survive?
Hopefully. I would love to see that. Yes.
Anybody have any questions in the room or online?
Hi, that was an awesome talk. I was wondering, you mentioned that there was no hyacinth in the local Hawaii, and so that maybe could potentially be correlated with the fact that it had native fish. What role does hyacinth play for fish? Is it like a place that they can hide and lay their eggs, or what makes it different?
So we actually saw, go all the way back. It's a really tiny picture, I'm so sorry. But my big head in that photo, all the hyacinth that you walk through, it basically sucks up all the oxygen that's like underneath it and around it. So we had traps placed underneath it, and then when we went to go collect those traps, we're taking the water quality. And in some locations under the hyacinth, it's literally reading like 0% oxygen. So a lot of the times the fish just like avoid it. So that's why the small fish seem to be thriving underneath it. Oh, probably. It's beautiful though.
Okay. All right. I guess we're going to move on then. So we're going to be going back out, thank you, to the Western Pacific, where Caroline is out in Ponape. I hope she is online. If you were there, Caroline, come back on. There you are. So Caroline is another rising senior, but majoring in geography and environmental science at Hilo. She's been mentored by Ashley McGuigan of the Southwest Climate Hub and Ryan Longman with his PDKE hat on, finding examples of sustainable water management practices across the Pacific to share with constituents. So you should have the helm. You should be able to grab and share your screen.
Got it. Oh, it says send request, so
I should just allow you. There you go.
Okay, can everybody see?
Yes, we can. I will do that.
Aloha, everyone. My name is Caroline Lynch, and today I'll be sharing my summer research as a surf fellow. My project focused on developing 2 new case studies for the Water Adaptation Techniques Atlas, or WATA, highlighting rainwater catchment systems in 2 Pacific Island communities. So the image here is a screenshot of the WATA website, which is where all the case studies are hosted. For context, WATA is an online tool developed to help communities, land managers, and water managers learn from each other about what works as well as what doesn't when it comes to water adaptation. It's a platform for sharing knowledge so people can try out techniques in their own contexts. While WATA has already a lot of case studies for the continental Southwest US. They've been working to grow their collection for Hawaii and the US affiliated Pacific Islands. So before my project, there were 9 Pacific case studies, which were all in Hawaii, and I was able to add 2 more representing the FSM and American Samoa. This photo here was taken at the Hawaii Conservation Conference with the Pacific Drought and Knowledge Exchange Team, or PDKE, which I had the privilege of being a part of. My primary mentor was Dr. Ashley McGuigan, and co-mentor Dr. Noah Silver Coates. So they supported me with the case studies. being part of PDKE, I got to attend weekly meetings and hear about the work that they do, as well as share my progress of my work with them. I was also able to attend a meeting with the Southwest Climate Hub and learn about the work that their team does as well. So this map here shows the Pacific region, highlighting Melanesia, as well as Micronesia and Polynesia, where my two case study locations are. In many island communities, especially low-lying ones like atolls, there are no rivers to draw from, so people often rely on rainwater catchment or thin groundwater lenses. And during droughts or El Nino events, these can dry up quickly, and climate change is making those events more severe as well. So studying rainwater catchment systems helps us to identify where water access falls short and find small upgrades that can make these systems more resilient. So on the left image is Bakin Atoll's Nagalap Island, and on the right is Tutuila Island in American Samoa. And these locations are very different. One's a more remote atoll community, and the other a volcanic island with more infrastructure. But they both do rely on rainwater catchment systems. Bakin's case study looked at storage reliability and drought resilience, while American Samoa's focused on water quality and public health risk. So this slide kind of is just a visual snapshot of my workflow, from reading the original research articles to drafting my case study summaries to them being published on WADA. So my role was kind of translating these dense technical research into something that's more practical and easy for people to use. This image here is from the original study for Bakin, and it shows the tanks on the island, which is like a mix of concrete, fiberglass, stainless steel, as well as PVC pipes. The research found that tanks often ran dry during droughts, but with larger roof areas and by implementing new tanks into service, the community could maintain a more reliable supply. Relatively small changes can make big impact in these kinds of situations. In American Samoa, 14 household systems were tested for microbial contamination, and 97% of those exceeded safe drinking standards. This was largely due to open tanks, no first flush diverters, as well as infrequent cleaning. So the takeaway here is kind of clear, that rainwater is very vital in these islands, but it's unsafe without proper treatment and maintenance. So this here is the WADA map with the QR code that links you directly to the site if you want to check it out. So the two new points on the map represent the FSM and American Samoa case studies that I added. So like I said before my project, all the Pacific case studies were from Hawaii. And by adding examples from the US affiliated Pacific Islands, we're contributing to help ensure the diversity of contexts as well as challenges in the region that's reflected. This matters because the issues here are often different than the issues we see in the continental Southwest case studies. So looking forward, I think that more Pacific Islands should be included in WADA to provide a broader set of lessons. Combining both quality and quantity assessments would probably give a fuller picture of water security, as well as keeping WADA updated ensures communities have access to examples that are relevant to their environment and challenges. I want to thank my mentors, Dr. Ashley McQuiggan and Dr. Noah Silber-Coates, for their guidance on my project and creating the case studies. I also want to thank the SURF program and Dr. Rachel Lentz. And I want to thank the USDA Southwest Climate Hub, PICASC, PDKE for allowing me to be a part of their team for the past 10 weeks, and Hawaiʻi Sea Grant for supporting this project. Mahalo.
Caroline, does anybody have any questions? I've got one. So in the process of looking at all of the examples that were already on the site, did you find that there were things that you think might help out in the USAPI? Or does it really need to have, do we need to have more studies specific to those islands to come up with some processes that might help them, do you think?
Yeah, I think there's probably a lack of research for the US API in that kind of sector. So definitely more research needs to be done before like people can go in and assess what they need and what works for them.
Okay, so some more work to be done in the future. Good. Anybody online or in the room have any questions? Nope. Well, we appreciate you taking time so far away from us to dial in and let us know about your work. Thanks very much.
Thank you.
Okay, let me reshare. Okay, so we are on to our last but not least. So Sophia Woofter is a rising junior atmospheric sciences major, there's your mic, at UC Berkeley. She's also been mentored by Ryan Longman, but this time with his Hawaiʻi Climate Data Portal hat. He wears lots of hats around here. And unfortunately, he's sick, so he's just seeing us online. She's been learning all sorts of stuff, but focused one part of her project on air temperature anomalies as a case study for using the HCDP by anybody out there. So show us what you did.
Is this clicker like good to?
Yes, that should work. In theory, it should work. We'll try with that.
Okay, great.
Yeah.
Okay. Hello, everyone. I just want to start off by saying thank you so much for your time and attention. I know it's been a long afternoon of some great presentations, so let's just finish strong. My presentation is called Climate Encoding. I analyzed trends in air temperature and worked with the Hawaiʻi Climate Data Portal on some website design. So I have some HCDP stickers circling around. Please take one and pass it down. So a network of weather stations across the islands collects data that the HCDP analyzes and translates into maps and tools that are easily accessible for researchers, students, and anyone interested in Hawaii's climate. I worked with HCDP over the past 10 weeks on some qualitative and quantitative research, so I'll get started on the qualitative end. I spent my time updating and designing the HCDP website using applications like Zotero and WordPress, and also did some outreach with Instagram and listserv. So Zotero is a literature archive program that I use to manually add over 300 new research publications to the HCDP library. So now this information is easily accessible and readily available for data portal users. I also used WordPress, which is a website design program, and spent my time on the cultural resources tab on 2 pages that weren't really touched since 2021. So I just revamped them with some pictures and new formatting. The first being the Indigenous Drought Perspective page, which discusses the importance of incorporating Indigenous and local knowledge into drought assessment and management across the islands. And the second being the Climate Glossary, which is just a collection of frequently used climate vocabulary and their translations in Olelo. I also assisted in weekly social media posts. You can see them at High Climate Portal. And I also created a listserv with over 1,200 subscribers, which is basically a mass email list. So now the HCDP can easily inform users about new data products that are continuously coming out. So now onto my more quantitative side of my research. I analyzed Hawaii's air temperatures over a 36-year period, Some skills I gained from this were learning how to code in the language Python on the program Jupyter Notebook, as well as some statistical analysis. So my research questions that drove my project were: first, can phase changes in El Nino southern oscillation explain variations in temperature? And are we seeing greater warming in recent decades relative to the past? My method of approach was to first download map data from HCDP. I downloaded two types of data, the first being the recorded monthly air temperatures from 1990 to 2025. So that is 426 files of recorded monthly air temperature data. The second being the historical monthly air temperature averages. So that's just 12 files corresponding to the 12 months. After I downloaded this data, I wrote code in Python to calculate these air temperature anomalies, and then I analyzed them using some statistical tests. So what this air temperature anomaly process looked like was something along the lines of recorded minus expected equals difference, that difference being the deviation from the mean or the air temperature anomaly. And what this looks like with the map data is taking the January long-term average air temperature and subtracting that from the actual recorded air temperature in January 1990, the result being the January 1990 anomaly map. And if you're wondering what that looks like, it looks like this. And I'm so proud of it because it took me about 2 weeks to just get to this step with one map. So what we're looking at is the January 1990 air temperature anomaly map, these bluer regions indicating where it was colder than average, and these warmer bands indicating where it was warmer than average, gray indicating no difference or 0. So after I got this one map, I had to run a loop through my code to find the rest of the 425 maps. And once I did that, I could now answer my research questions. The first being, can phase changes in El Nino Southern Oscillation, or ENSO, explain variations in temperature? And I know I'm looking at a room of scientists, but just for some background information, ENSO is a reoccurring climate phenomena associated with either warmer or colder than average sea surface temperatures. And it's very relevant to Hawaii's climate because this phenomena takes place only in the Pacific Ocean. So we see El Nino phases associated with these warmer than average temperatures, while the La Nina phase is the cooler than average temperature. So how I compared the anomalies to their ENSO phases can be seen in this box plot. What I did was I separated my anomalies by the ENSO phase they were in. So as you can see, anomalies in El Nino and neutral phases, their median line was around this 0.1 degrees Celsius mark, and La Nina anomalies were around this negative 0.14 degrees Celsius mark. Even though this is a nice visual, it's important to test for statistical significance. So I ran a normality test on this data and found that this was not normally distributed, and that's okay. It just means that I had to run a non-parametric test for significance. Upon doing that, I found that anomalies in El Nino and La Nina phases were significantly different, as well as anomalies in La Nina and neutral phases. However, the test could not detect a significant difference between the anomalies in El Nino and neutral phases. What this actually means is anomalies in El Nino phases were significantly warmer than La Nina anomalies. La Nina anomalies were significantly cooler than neutral anomalies, but El Nino anomalies were not significantly warmer than the neutral anomalies. With my first research question answered, we can now move on to the second. Are we seeing greater warming in recent decades relative to the past? So what you're looking at is a histogram showing the distribution of the anomalies over the data set. On the x-axis, we have the anomaly value, and on the y-axis, we have the frequency, or how often this value occurred in the data set. To get an idea of the greatest warming, I calculated the 99th percentile, or the top 1% of this data. The threshold for this was 0.68 degrees Celsius, meaning any anomaly greater than or equal to that value was considered an extreme anomaly in that top 1%. So in that top 1%, I found there to be 45 anomalies, and This is a bar chart of those 45 anomalies. On the x-axis, we have the years they were in, and the y-axis, how many months of these frequent anomalies there were. As you can see visually, there seems to be a higher amount of these extreme anomalies in the latter half of the data set. But again, to test this for statistical significance, I created another box plot. I split the anomalies in half by their years. So this is the first 15 years. This is the latter 16 years. You can see the median lines show that the latter half of the data set does show that there are more extreme anomalies in that half. But let's test for significance. I ran the same normality test, found that again, it was not normally distributed. So I ran the same non-parametric test for significance. And indeed, these two boxes are significantly different. And what this means is extreme warming is significantly more frequent in these past 16 years than in the previous 15 years. So now with my research questions answered, we can circle back and make some conclusions. The first, can phase changes in ENSO explain variations in temperature? Yes, my test did prove that anomalies in El Nino and La Nina phases were significantly different, as well as anomalies in La Nina and neutral phases. However, there was no statistical significant difference between anomalies in El Nino and neutral phases. Second, are we seeing greater warming in recent decades relative to the past? Yes, we are seeing this higher frequency of these extreme temperatures more frequently in recent decades than in past decades. Some next steps for this project would be to determine if it is ENSO that is driving this higher frequency, or if it is a result of anthropogenic warming, also known as climate change. This would require a lot more time and coding, so it's a future project, but it is one that I'd be interested in pursuing. And now I'd like to say very many thanks. Thank you first to my amazing mentor, Dr. Ryan Longman, not only for his guidance and support, but also for just like giving me so many experiences over these past 10 weeks. This is a photo of us at the Hawaiʻi Conservation Conference. I'd never been to a conference before, so that was really cool. I also got to see a weather station, see a thesis dissertation, and attend like weekly meetings. It was just so fun. Also, thank you to Shariel Hugh and Dr. Carrie Kodama in the back. They taught me how to code. I had zero coding experience before this, so I'm very grateful to them for setting aside time in their busy schedules to meet with me and answer my questions. Also, thank you to Amy Schreiber, who is also in the back. She taught me how to use Zotero and WordPress. I, again, had no skills in that, so very grateful to Amy for teaching me. Also, thank you to Dr. Tom Jambaluka and Maddie Lucas for supporting me during weekly meetings and just welcoming me to HEDP with open arms. With that, thank you.
Oh, you learned so much.
I didn't know I would be able to fit it all on one presentation.
Excellent. Do we have any questions in the room or online? Everybody's talked out. I know. Oh. Go ahead.
Thank you very much. Excellent talk. Wonderful. I'm going to get a good question. Do we know, and maybe you said it and I missed it, but do we know about ENSO habits, behavior, whatever, longer than just this last century or the data that we have? Like, do we know more about it over longer periods of time?
Yeah, that's a good question. I did. So how I got my ENSO data for those anomaly months was I looked at the ONI index. It's provided by NOAA, and at least from what I remember, they have data from back to 1930. So yeah, a much larger time frame than what I was just looking at. But the HEDP data, air temperature data I was using started in 1990.
I sort of have a vague memory, this might be wrong, but that there might be some references in the Hawaiian newspapers that have been interpreted as potentially being El Nino events. Is that true?
Yeah.
where they measured, oh, thanks. Yeah, there's a bunch of detailed reports from the plantation era where they were reporting like daily temperatures. I can't remember exactly from when, but beginning in the early 1900s, and then in the Hawaiian newspapers in terms of like phenological phenomena, like the arrival of specific fish species and things like that can be interpreted as potentially being correlated with El Nino events. I think Justin Suka's been working on that. So hopefully we'll get to hear about that work soon because I think they're finishing that project this semester.
Because I think there was an early PyCast project that Rosie was doing that combined the newspaper work as well as tree ring work to look, because the thing is there aren't a lot of trees in Hawaii that have rings, but there are some of the Norfolk pines. So she was doing some dendrology. That's what's called, right? Yeah, so I think there are signs that we have information from before documented, whatever. Any other questions? Anybody online who has questions about any of the talks, really? It's a free-for-all. No, we're good. Are we saturated? Uh-huh. It was. I think this is the thing that I find remarkable. There are very few of these students who came in with knowledge about any of the topics that you just heard about today. And you can see how much they have learned and how much they have done to contribute. I'm always amazed at how much they absorb over just 10 weeks over the summer. And it's so cool. It is so gratifying. And I think it's really what helps drive doing this program, because we love to see students learning new things and then taking that and going beyond. As I mentioned, one of the new things we did this summer was a workshop with SURF alumni. So we had 4 alums come back and talk to the students about the paths that they have taken since graduation, and they were all very different. And it was a really neat event. So I'm hoping that we can, the cohort is building and it'll be a great alumni network these people can tap into. You can always come back and talk to us at both PyCast and Sea Grant. So with that, I should probably say, you can step down. And I will unshare. There we go. We love seeing all of this great science, especially now. We wish you the best of luck in your scientific careers. Keep at it. Thank you for working so hard over these 10 weeks of the summer. I hope that you enjoyed it. We wish you the best as you continue in your journey. And we do ask you to please keep in touch. I will be periodically sending out emails to alumni, and I hope you will answer. A very big thank you to their mentors. We cannot do this without mentorship. And you've all done a tremendous job this summer, as past ones have. Make sure to tell your friends that you had a great time with a surf student this summer, and that they should also volunteer next year to have a surf student, and it will be worth their while. I wanted to check and see, I know that there are some leadership who are online, or at least they were supposed to be. There's one in the room. If any of you folks would like to say, that would be you, Darren. Oh. Our leader, Darren Lerner. And I think Ryan is online as well. I don't know if Mary Vaughn is. I haven't looked at the list of people. But if any of leadership would like to say a few words at the end of this event, please unmute or stand up with a mic. There's Ryan.
I have the green light. I have the green light. All right, sorry I couldn't be there today. Definitely a bit under the weather, but really amazing research. I watched every presentation with enthusiasm. I'll just mention that when we started the interview process, my concern, we couldn't even figure out who we were going to select. Everybody was such a strong candidate that it was really surprising, and I just knew everyone was going to succeed. And to see what kind of research you're producing for someone that's been around for a couple of decades now in this space, it's really outstanding to see a student go 10 weeks and pull together that kind of research, and presentations were excellent also. But I can go a little about the students, but I think what I really came online here to do is to acknowledge the effort that Rachel Lentz had put into this effort. She has gone above and beyond to make sure that everybody was paired with the right mentors, and organizing those extra activities. And really, I think the success of this program, good students means it, but also good organization. So can we please give Rachel a hand right now? I can't hear the applause, but I'm hoping people are clapping. So on that note, really thanks to Podcast and Sea Grant for funding this opportunity. I had an absolute pleasure working with both Caroline and Sophia. I learned a lot, and their enthusiasm inspired me to keep doing the work that I do and to work with more students in the future. So thank you very much.
Great. Taryn, do you want to come up here?
Well, I don't think there's too much more I can add to what Rachel and Ryan has said. I think a round of applause for all our students again, if you wouldn't mind joining me in that. Ryan, of course, captured appropriately the thank you to Rachel for running this program. And how many students did we have the first summer?
The first summer we had 3.
We had 3.
But it was COVID. So
Yeah.
they were very brave souls to take that on.
Yeah, so 3, and we've been growing ever since, or at least trying to grow, and then we really have kind of taken a jump here, and we hope to continue that. So, you know, I'm searching for the things. Well, I guess I should say, if I don't know you, I'm Darren Lerner. I get to serve as the director of Sea Grant. I've served in Ryan's seat with PyCask. I'm now, I guess, executive director with PyCask. Ryan, if we didn't get his title out, is our program director for PyCask. I think adding to what Rachel said, though, or maybe underscoring it, she said, you know, we really hope that you'll, you know, respond when we email you. We want to stay connected. I think more than that, we are here for you. You're part of this Ohana, and that's for always. So anytime you want to reach out, you're finishing your undergrad, you're looking at maybe going to graduate school, jobs, whatever it is, there are a whole bunch of people here that are super happy to help you. So please don't hesitate to reach out. A thank you again to Rachel for taking on quite a bit of work to run this, and quite a bit of wrangling with those professors trying to get them on board. Although good to hear that this year we had a lot of people. And I think that's part of it, is we're building some momentum here, and you're part of that story of building that momentum. And as we do that, and as such awesome students come on board and come and go, word gets around, and we have more and more faculty that step up to be those mentors for you. So Rachel said it, we just wouldn't even be here if it weren't for all of the students. So thank you so much. Really appreciate it.
And we are done. So, oh, we're going to have a picture. So don't go anywhere. And I guess if we could have, if Caroline and Bianca are still there, we could have them with their cameras open and we can put them behind. All right. Otherwise, everybody else, thank you for coming, and aloha.